A very big hello to all my wonderful PU1 champions. I hope you're doing great. Yes, so today we are going to solve the model question paper, and by if any chance, if at all you are watching this video for the first time, let me tell you, you can do it. Okay, you can do it if at all you have not prepared really well. Trust in yourself, put in your hard work. I'm telling you, you can do it. Just work hard. Okay, and smart also. So we'll make sure that the whole model question paper is complete and you understood everything well, then only I'll be leaving this particular place, right? And my dear children, I just want one thing from you. Listen to me very carefully while I teach. Do not make notes as of now. Just grasp everything which is in there. Probably after solving one part A, you can pause the video and just go through it if at all you want. That is according to your wish. Yes? So let's go ahead with the first part, that is part A, 1 marker, and over here you have the first question. In the hierarchical classification, order is placed between. And the trick that I'm going to tell you, I'm so sorry, this is what I have been telling for almost so many years now. So please forgive me if at all any of my old students are watching this. So the way I remember the hierarchical classification goes like this. King Peter came over for a, we do not want A, okay, for a great soup. So King Peter came over for a great soup. That is how I remember. K stands for kingdom, P stands for phylum, C stands for class, O stands for order, and F stands for family, G stands for genus, and S stands for species. That is how it goes. Now, if at all you have any other fun mnemonics to tell me, let me know in the comment section below right away so that your teacher can be upgraded. Yeah? Okay. Now, you are being asked, where is order placed? Order, if at all you see, it is placed between class as well as family. Class and family. Do you have an option like class and family? No, I have family and class. Thus, is it okay? Yes, it is okay. You do not have any other closer options. So family and class will be okay, even though it's in different order, but it's okay. Family and class. Fine, done, deal. That's all. Easy question. Now, next thing. Which of the following is the correct sequence of class, mycelium, fruiting body observed in the kingdom fungi? So now we are entering into kingdom of fungi, and my dear children, you need to understand that fungi is made up of one very important thing which you call as mycelium. But do you know what mycelium is made up of? Mycelium is made up of another very important thing which is known as hyphae. What do you call this? Hyphae. Now, there are two cases. This hyphae which is there, it might be divided like this. Whenever you have a division like this, you call this condition as septate condition. Okay, septate condition. Now, if at all these divisions are not there, what do you call this as? Now, we will not call this as septate. Everything went off. Now, we will not call this as septate. Rather, you will be calling this as aseptate. Aseptate, that means septum is not there. Okay, and also usually in aseptate condition you could see one more very important thing, that is the nucleus which is there. There might be multiple nucleus out there, and this is known as cenocytic condition. What do you call it? Cenocytic condition. So cenocytic condition can be seen, and this is mostly associated when the hyphae is aseptate. Okay, done. Now, you have been said that you have to find the correct ones, which has been correctly matched. Okay, Phycomycetes have septate and cenocytic mycelium. Do you think it is septate and cenocytic mycelium? The answer is wrong. Actually, they have aseptate. Okay, they actually have aseptate mycelium, not septate one. Okay, and fruiting bodies is actually not present in them. So basically, I'll say this particular portion is correct, but over here, if at all you see this is wrong, which is why this is definitely not the correct option. Ascomycetes or mycetes, okay, ascomycetes. Here you have aseptate condition and branch. Listen to me carefully. 3 of them that you see over here, all of them are separate. Only one guy was aseptate, and that was Phycomycetes. So basically over here aseptate is given, so this is wrong because this is actually septate. Here also aseptate is given, that is also wrong because that is also actually septate. And over here septate is only given, so that could be correct. So Deuteromycetes, my dear children, they are septate and branch and the fruiting body is not present. correct. Okay, correct. So 3 of them, always remember, they are having the septa, whereas phycomycetes do not have the septa. They are aseptate in condition. Will you remember that? And since here everything is correct, the answer for this particular question is going to be D. If at all you want to read it, you can read it. So phycomycetes, the mycelium is aseptate and sinusitic. Ascomycetes, mycelium is branched and septate. Basidiomycetes, the mycelium is branched and septate, just like this. Yes, just the fruiting bodies are going to be different. Here it is azocarp and here it is going to be basidiocarp. Cool. And here in deuteromycetes, it's not present only. Fine. Now, which of the following belong to class Osteichthyes? What do you mean by Osteichthyes? You know, fishes which are there are 2 types. One, which have their endoskeleton, okay, endoskeleton. Inside our body, we have a skeleton, right? So the endoskeleton can be made up of bones or it can be made up of cartilage. For example, over here, when we look at our ears, it is made up of cartilage, which is why you can do like this. Yeah, similarly our nose, we can do like this and like this. Don't do at home. Yeah, you can try it out at home. It's not harmful. Yeah, so basically all of these places where you can do all this drama out there, it is made up of cartilage. Cartilage is considered to be brittle in nature, and basically it can change its shape like this. Okay, not shape exactly, but yeah, we can move it very easily. So if at all I talk about fishes, they are also like this. They have their endoskeleton either made up of bones, and the ones which have bones as the endoskeleton, you say that they belong to class Osteichthyes. Whereas there are fishes whose endoskeleton is made up of what? Cartilage. In that case, you call them as, you say that it is under class Chondrichthyes. What do you say? It is under class Chondrichthyes. So C for cartilage and C for Chondrichthyes, you can remember like that. So out of here, you have been asked who all them have what, who all them come under Osteichthyes and who all them basically have bony endoskeleton. That is the question. So Scoliodon, my dear children, do you think it has bony endoskeleton? Scoliodon is known as dogfish and it has actually cartilaginous endoskeleton. Yes. Okay, what about Exocetus? Exocetus has bony endoskeleton. Hippocampus also have, this is seahorse, my dear children, this also have bony endoskeleton. Okay, Carcharodon, what do you think about them? What is this? Shark. Now shark, obviously ma'am, it's a very tough organism, it will be having bone. No, it has cartilage. It has cartilage. So obviously that is also odd one out. What about Clarius? Clarius, my dear children, has bony endoskeleton. So basically if at all I see 1, 2, 3 of them, that means 2, 3 and 5. 2, 3 and 5. 2, 3 and 5. These are the ones which will be having bony endoskeleton. Okay, so Osteichthyes are bony fishes with a skeleton made up of primarily of bone. Example, Exocetus. Exocetum is what? Flying fish out there. Hippocampus is the seahorse and Clarius is the catfish. All of them has bony endoskeleton. What about Scoliodon? Scoliodon is a dogfish and Carcharodon is basically giant white shark, great white shark out there, and that is going to have cartilaginous endoskeleton. You say that they come under Chondrichthyes. Okay, Chondrichthyes. Yes. Perfect. Now read the statements with regard to types study. Okay, anyways, study of frog and select the correct answer from the below given options. Okay, we have to find which one of them is correct about a frog. Okay, vasa efferentia are 10 to 12 in number that arises from the testis. What is vasa efferentia? It's a duct that carries the sperm. Okay, and here it is said that they are 10 to 12 in number. In the case of frog, what do you think? Are they 10 to 12 in number? The answer is yes. So there is no point of discussion over here. It is a factual thing. 10 to 12 is there in the case of frog, and that is vasa efferentia. Okay. Now the medulla oblongata passes through the foramen magnum and continues as spinal cord. So basically, listen, this is your brain, right? So your brain which is there, it has different portion. There is forebrain, okay? There is hindbrain, and in between that you have the midbrain. The hindbrain, okay, the hindbrain which is there, it has different parts. Cerebellum is there, you have, pons, you have medulla, all of this thing. So here it is said that medulla oblongata, which is a part of the hindbrain, the last part of your brain, passes out through foramen magnum. What is foramen magnum? So basically, it's an opening in your skull. Now I'm relating it to myself, but actually it should be answered about frog. So frog also, my dear children, they have an opening in the skull through which the last part of brain is going to pass through. Just imagine, this is what your medulla oblongata, and And then if at all you go further, you will be finding what? Spinal cord. So this is like a connecting link that you have. The medulla oblongata which is there, it is like connecting link that you have towards the spinal cord. Medulla oblongata belongs to your brain. It controls major things, for example, your respiratory rate out there, your heartbeat, your blood pressure, everything. It's super important thing. Okay, and basically it passes through this foramen magnum. And over here I have a picture of it. This is not a frog. I have clearly written over here just to give you an understanding of how this opening is. Okay, cool. So this is also correct, my dear children. Now frogs are uricotelic. Uricotelic means their nitrogenous waste is uric acid. Do you think their nitrogenous waste can be uric acid? What do you think? You know, uric acid is found in the case of, lizards and things like that. In them, there is no fluid that is being secreted because their body, my dear children, would not be having too much amount of, they do not have too much amount of water to waste off. Okay. So in that particular condition, it will be a very dry thing that is going to be released as waste. So we say in our case, my dear children, we have urea as the nitrogenous waste. But for urea to move out of our body, since it is toxic, basically, since it is toxic, you need to dilute it with some water, which is why water also moves out, which is why it is water also that is moving out in our case. Okay, so in there, we are ureotelic. Similarly, over here, when you talk about lizards or birds, they do not have much amount of water to waste, which is why they have a less toxic nitrogenous waste, which is known as uric acid, and that does not need too much amount of dilution, which is why it can be just dry thing which is there. The white thing that you see is uric acid. Okay, if at all you have noticed. Now, talking about frog. Frog lives in water as well as on land. It does not have any problem with having water out there, right? Or removing some amount of water out there. So do you think they can be uricotelic? No, they are not uricotelic. Only the organisms which are very much struggling with loss of water, they would be the ones which would be uricotelic. So this particular option is definitely going to be wrong. Now ovaries have no functional connection with kidneys. Ovaries for production, kidney is for what? Excretion. So obviously there is no functional connection over here. So that is correct. So basically, can I say that 1, 2 and 4 statements are correct? 1, 2 and 4 statement. 1, 2 and 4 statements are correct, while statement number 3 is what? Wrong. Why is it wrong? Because frogs are ureotelic. Just like us, frogs are also ureotelic. They are okay with removing the nitrogenous waste with some amount of water. Okay, they are ureotelic, not uricotelic. Going ahead, which of the following given options describe the graph correctly? So over here you have a graph where the potential energy is given over here and there is reaction that is happening over here. If at all you see this graph properly, there is substrate and basically there is product. And if at all you see the graph properly once more, you will be able to find that the energy has increased. The energy has increased, then it is decreasing. That means it was high here, it became low here. It was high here, it became low here. So when the chemical reaction happened, probably some amount of energy got lost when the substrate converted into the product. That means this type of reaction, when the energy is lost, what do you call this type of reaction as? You call this as exothermic reaction. What do you call this as? Exothermic reaction, because the energy is being released. If at all the energy was more, the graph would have went up, and my dear children, those reactions which requires energy, they are known as endothermic reaction. So over here, 2 options which are there, they are endothermic, so I'll cancel them off. I do not want to see those options. Let's go ahead with exothermic options only. Now, the next question that you have is, so exothermic reaction with energy, A, in the presence of enzyme, and B, in the absence of enzyme. What do you think? What is this? So here, if at all you see, there is a spike in energy. There's too much spike in energy over here. What is this energy depicting? What is this energy called as? This is known as activation energy. Activation energy is basically the energy required to initiate the chemical process. So whenever you have an enzyme, the energy required to initiate would be less. Whenever you do not have an enzyme, obviously you would need too much amount of activation energy to initiate the process. So basically, can I say over here, since too much amount of energy is required, can I say this is without enzyme? And this one would be with enzyme, since here activation energy is less. So A is with the enzyme, A in the presence of enzyme, and B in the absence of enzyme. So this is an exothermic reaction. Why, Baba? Because the energy graph is going low, and here it is having less activation energy, and here it is more activation energy, which is why I'll say that this happens in the presence of enzyme and this happens in the it happens in the absence of enzyme. So my dear children, your answer for this question is going to be B. Yes? By the way, are you understanding? What? Are you understanding? If at all you're understanding, if at all you're liking this video, what do you do? You have to subscribe to the channel. Yes, yes. So activation energy is the minimum amount of energy required to start a chemical reaction, and that will be less in the processes involving enzymes, because enzyme will do the job. Okay. Now question number 6. Identify the substages of prophase I with respect to the given features out there. So this is not in order, I'm telling you. Okay. This is not in order. You just have to identify what is happening when. Are we ready? Okay. The thin thread chromosomes with a beaded appearance happens when? In leptotin stage. In leptotin stage. So I say the chromosomes basically appear in leptotin stage. Okay, fine. Appearance of recombination nodules. Let's just keep it there. Okay. Formation of bivalent and tetrad. What do you mean by that? The two chromosomes which are there, the homologous chromosomes, they come together, and this is known as bivalents or tetrads. Okay, bivalents or tetrads. Why? Because one has two chromatids, one also has two chromatids. In total, tetrad is something that is being formed. So formation of bivalent tetrads happens in which stage? This is known as zygotine. And now, when they are very close to each other, there are chances that crossing over is going to happen. Okay, so there is appearance of recombination nodules. There is appearance of recombination nodules that happens in the next stage, which is known as pikitin. Okay, fine. Now, after that, until now, okay, when recombination nodules are being formed, there is a synaptonemal complex being formed, my dear children. There is something that is gluing both of these bivalent things, chromosomes out there. Now, whenever you want to separate it, first of all, the synaptonemal complex in between is going to go off. But still, the chromosomes are going to be attached at some point. This is known as chiasmata. This is known as chiasmata. So, appearance of chiasmata happens in the next stage, which is known as diplotin stage. And further, you want them to move away. So basically, chiasmata which was here will also move away, go towards the end. So that is known as terminalization of chiasmata, and that happens in dikinesis. Dikinesis. The last stage of our life is what? To die. So that is why it is also will be last over here. So if at all you see by that nature, my dear children, what would be your answer? One is going to be leptotin. One is leptotin. One leptotin. Okay, everyone is leptotin. Fine. Second one is pikitin. Second one is pikitin. Okay, I got two options. Let me cancel these two options. Just a second, let me cancel these two options. Okay. The third one is zygotin. And the fourth one is dikinesis. Okay, and the fifth one is diplotin. Yes. So your answer for this question is going to be C. Okay, and there is a way to remember it again. Lazy zebras play disco daily. Okay, lazy zebras play disco daily. L for leptotin, Z for zygotine, P for pikitin, D for D for diplotin, and this is dikinesis. Okay, dying comes at the end. You remember like that. And over here, I have just mentioned the order and the things that happens in this thing. Okay, it's not like 1, 2 goes with 2 there. No. I'm just explained to you in the order so that if at all you want to just have a quick read of what happens in what stage, you get an understanding according to the flow, actual flow of cell division. Okay, fine. Now, ATP and NADP is produced in the light reaction of photosynthesis by the movement of electrons in ETC, that is electron transport chain, are used immediately for. So there is what ATP and NADPH being produced. Where do you think they are used for? Where do you think they are used? So in the light reaction they are being produced, but actually they are produced in light reaction for being used up in the dark reaction. For being used up in the dark reaction. What is happening in the dark reaction? There is reduction of CO2, carbon dioxide, to what? To basically your carbohydrates. To the carbohydrates. Or do I call it as sugars? Reduction is happening, and in this process you require too much amount of ATP as well as NADPH. Well, this is also known as biosynthetic phase. Dark reaction is also known as biosynthetic phase. Why? Because lots of new things are being synthesized right up over here. So where are they used up? While oxidation of carbohydrate is not happening up over here. Synthesis of sugar in the biosynthetic phase, yes, that's when it is used up, and it is also known as reduction of carbon dioxide. When reduction of carbon dioxide happens, that's when you get the carbohydrates. So can I say both B and C options are correct? Yes. So which is why my answer for this question is going to be D, both B and C. Done? Done, ma'am. Okay. Next, refer the given equation. The equation is given like this. Are you able to see it? If not, I'll just zoom it in a bit and I'll show you. Yo. Yeah. Now, able to see it? You have to find the respiratory quotient in this particular case. How do you find the respiratory quotient? Any idea? There is a formula. And if at all you do not know the formula, you should know the formula. It is the ratio of volume of carbon dioxide produced to the ratio of oxygen utilized. Okay, and if at all you do that, so basically volume of CO2 produced divided by volume of oxygen being consumed or being utilized. Okay, so if at all you see that over here 102 CO2 molecules are being produced. So 102 divided by how many oxygen molecules are being utilized? 145. So 102 divided by 145, your answer is going to be around 0.73. How do I remember? Because 0.703. So basically, how do I remember? Because I've already seen it, right? So I've only made the PPT. So over here, your answer for this question would be 0.7. You do not consider the 0 over here. If it was more than 5, you would be still considering it, but still, since it's 0, you do not consider anything. Your answer would be just 0.7. Okay, got it? Why is that answer not marked? I'll mark it for you. Okay. Now going ahead, efficiency index in the geometrical growth is the ability to produce what? So what is efficiency index in geometrical growth? Geometrical growth means exponential growth. Cell is dividing, increasing exponentially, 2 to 4, 4 to 6, and goes on like that. So the efficiency index in geometrical growth is the ability to produce what? It is basically, efficiency index is the term used in the case of plants. So it is just, you just have to remember, it has something to do with the increase of cell number. It has something to do with the increase of cell number out there in the case of a plant. So here it is written, is the ability to produce cell wall? No, cell wall has nothing to do with the growth. You're talking about growth over here. Cell wall formation happens, okay, but the cell is not dividing. Only if the cell divides, that's when growth is going to happen, right? New enzyme formation, again, has nothing to do with the growth. New plant material, yes. New plant material means some growth has happened, something has been formed up there. Young ones through mitosis, again, over here it's not too much specific. Here it is clearly mentioned as plant material, so we will go with this one. New plant material. So efficiency index in the geometrical growth is the ability to produce new plant material or to have more growth in the plants. That's all. I know this is an off topic out there, but yeah, okay. Now question number 10. During expiration process of breathing, what happens? During expiration, you are expelling out the air from your body, that is expiration. When this happens, thoracic volume increase. So my dear children, just imagine, the diaphragm is like this. Okay, when it is like this, we say it is contracted. Yes, I know it might seem very odd to remember it that way, but yes. When the diaphragm is flat, I say that it is into the contracted stage. When the diaphragm is going to move up, I'll say that it is now relaxed. Just like how when we are like this, we say we are relaxed. But when we stand, we say it's like, oh, it's difficult, right? So that's the exact thing that is happening up over here. So diaphragm which is there, it is actually contracted over here. And now when we breathe out, this moves up, becomes dome shaped, and it is relaxed. So over here it is mentioned that diaphragm is contracting. Wrong. It is relaxing. The second thing, thoracic volume addition. When this goes up, whatever space was here, it is getting less, right? So can I say thoracic volume decreases instead of increases? Obviously. So this will also be wrong. Now, intrapulmonary pressure increase above the atmospheric pressure. So if at all you see, within the less thoracic volume, all the air is still stuck up there. You get it? So obviously the intrapulmonary pressure over here in the lungs, my dear children, the pulmonary pressure is way too much, and it is more than the atmospheric pressure out there. That is why expiration is going to happen. That is why expiration, from more to less it will be going, right? So that is why expiration is going to happen. This is why I say that B is correct. Now sternum is present at normal position. So when we expire, when we breathe out, do you think the sternum, what is sternum? Sternum is this. onto which your rib is going to be attached. You call this as chest bone also. So when we breathe out, so first of all, when we breathe in, all of you touch like this and breathe. So when I breathe, it goes up. When I breathe out, it goes back to the normal position. So this is also correct, that sternum is present at the normal position. So can I say both B and C options are correct? Yes, ma'am. Yes, that's all. So your answer for this question would be D. Cool, cool. Now next one. Arrange the given steps of human respiration in the sequence they occur. Okay, first read out all the options carefully. Diffusion of gases, oxygen and CO2 across the alveolar membrane. Alveolar membrane. So basically I'll tell you the flow. Okay, we breathe in, all the gases goes to the alveoli. From the alveoli, the gases are going to diffuse into the blood vessels. From the blood vessels, it goes to the rest of the body. In the rest of the body, oxygen is being transported inside, CO2 is being taken back. That is how it usually goes. Okay, so diffusion of gases O2 and CO2 across the alveolar membrane, this is definitely going to happen. Before that, we have to take in the breath. Is that point mentioned over here? Transport of gases by the blood, utilization of oxygen, pulmonary ventilation. Yes, pulmonary ventilation, by which atmospheric air is drawn in and CO2 rich alveolar air is released out. Yes, so atmospheric air which is rich in oxygen is being taken inside, and the CO2 rich blood, say CO2 rich alveolar air is going to be breathed out. Okay, correct. Now it's okay. Now we have taken in the oxygen, right? Now diffusion of, let's go ahead. Now, yes. Next would be this. Diffusion of gases O2 and CO2 across the alveolar membrane. So ma'am, we just released the CO2 right now, right? Then how does CO2 come back again? Listen to me carefully. So we have taken in, now in the alveoli you have oxygen. In our blood, still CO2 is there, which has to be removed. So oxygen will be going inside. CO2 which is there, it goes into the alveolar. Because the next process will be breathing out. It will be going out then. Okay. So diffusion of gases O2 and CO2 across the alveolar membrane done. Now what happens? Transport of gases by the blood. So this was first, this could be second, and this could be third. Transport of gases by the blood, and then utilization of O2 by the cells for catabolic reaction. Okay. Before that, do you have something else? Diffusion of oxygen and CO2 between the blood and tissue. Yes. First of all, you transported it. Now again, when it reaches to the cells are needing oxygen and CO2 is given back. So that diffusion has to happen between the blood and the tissue. Yes. So diffusion of oxygen and CO2 between the blood and tissue. Now this oxygen which has been taken up will be utilized by the cell for catabolic reaction, for any metabolic reactions out there, for breaking down things that is basically in the process of respiration. For breakdown of glucose, you require oxygen. Okay, and then when you do this reaction, again CO2 is going to be released and the CO2 has to move out. That is how generally things work. So your answer for this question is going to be what? Let's see. So the first one is fourth option. First one is fourth option, and you just have one option like that, which is why your answer is going to be like this. Go through this once more. If at all you want to pause the video right now, you can pause it. Done. Okay. Next one. Match the following column 1 with column 2. Okay, column 1 this, column 2 this. Superior vena cava, inferior vena cava, pulmonary artery, pulmonary vein, and what do they function? That is the thing. So always remember, your heart is what? Four-chambered in structure. This is left atrium, this is left ventricle, this is right atrium, and this is right ventricle. This is your lungs, and this is your body parts. From the lungs, we get oxygenated blood because we have just taken in. This oxygenated blood will go to the left atrium. Why not right atrium? Because left atrium is yelling la oh, bringing oxygen. You can also remember it as Los Angeles, whatever you want to remember. So it goes up there only, okay? And from here, it goes to the ventricle. From the ventricle, it is going to be pumped to different parts of our body. Now, always remember, whenever you say that blood is taken towards the body, it has to be a vein. So, sorry, blood is taken towards the heart, it will be a vein. Towards the heart, this is taking blood towards the heart, so it has to be a vein. And whenever you have lungs being involved, you use the term pulmonary vein. Now over here, blood is taken away from the heart. Away, A for away and A for artery. A for away and A for artery. So artery is the one that is going to take the blood away. But this artery has a different name, and that name is aorta. What do you call this as? Aorta. Fine. Now in the body, the oxygen is being given. Now the body will give what? So much amount of carbon dioxide. So I'll call this blood as deoxygenated blood because it has lots of carbon dioxide in it. Now this deoxygenated blood again goes to what? Atrium or ventricle? It first goes to the atrium. A comes before V, right? So it goes to the atrium. And then from the atrium, it goes to the ventricle. From the ventricle, where will it go? This is deoxygenated blood. It has to be reoxygenated. For that, it has to go back towards the lungs. Now listen to me carefully. It is this particular thing, whichever is there, it is taking the blood towards the heart. That means again it is a vein. And this vein, my dear children, you call this vein as vena cava. What do you call this as? Vena cava. Remember, it is deoxygenated blood which is being carried. Now there are two vena cava. Superior vena cava, which collects the blood from the top of our body. Inferior one, which collects the from the bottom of our body. So I say that vena cava is there, superior one and also inferior one. Fine, done. Now here, from the right ventricle, the blood is taken to the lungs, towards. It is basically away from the heart and towards the lung. Away from the heart, that means again it has to be artery. And whenever you have lungs being involved, I'll use the term pulmonary artery. Got it? So you have two pulmonary, here pulmonary artery, pulmonary vein, and here you have aorta, here you have vena cava. Now, superior vena cava brings what? Deoxygenated blood. So you not have to look at oxygenated. Let's look at deoxygenated blood. Deoxygenated blood from which part of the body? From the upper part of the body. Where does it bring to? It brings to the right atrium, right? It brings to the right atrium. So can I say the answer for this question is 4? Yes, ma'am. Inferior vena cava brings deoxygenated blood from the lower part of the body to the right atrium. Isn't it bringing the right atrium? Yes, Baba. So that is also correct. Now, pulmonary artery which is there, it carries the blood away from the heart towards what? The lungs. Pulmonary means it has something to do with the lungs. And basically, artery taking the blood towards the lungs. It has to be pulmonary artery only. Now, that goes with this. Carries deoxygenated blood. Pulmonary vein carries oxygenated blood. So what do you think is the answer for this question? This is your answer for the question. A goes with 4 and B goes with 3, and I have marked everything else. Done. Now, 13th question. Choose the mismatched pair of nephron with its function. So first of all, do you know the structure of nephron? It looks like this. You have a capsule which is like a bowl-shaped thing. You call this as Bowman's capsule. Okay. Then you have a twisted tubule. You call this as proximal convoluted tubule. Then this proximal convoluted tubule is going to make a structure like this, not this small, okay? Just chumma making. Like this. Okay, which is known as a loop. This is known as loop of Henle. So we'll just carry it. This is Bowman's capsule. This is PCT, proximal convoluted tubule. It is proximal to Bowman's capsule and it is convoluted. And here the loop that is being formed, it is known as Henle's loop or loop of Henle. Okay, and then you have the distal convoluted tubule. It is a distant and it is also convoluted, and together they open into what? A duct that is known as collecting duct. Now listen to me. This was DCT. So usually what happens here, you have blood capillaries, okay? Blood comes rushing here, and here the capillaries are there, they are very thin walled, which is why certain ions, okay, certain ions, your glucose, ions such as what? Sodium, potassium, all of it, okay, glucose, all of them, they come inside. And apart from that, the waste materials which are there, urea and all of it will be coming in. Not proteins, it can be amino acids that comes in, not proteins. Okay, so all of them are now passed into the Bowman's capsule. Why? Because of the pressure. Now where they are inside, and they came here because the capillaries were having small spaces, there was leakage that is happening. It's not like we did not need this glucose or we did not need these ions. Urea, I know we need to excrete it, but rest of the things we need it sometimes. So body gets a chance to reabsorb it. First of all, what do you call this particular thing as? When things are being moving like this. This is known as glomerular filtration. This happens in the Bowman's capsule. So this is correct. Glomerular filtration happens. Then after that, my dear children, what exactly happens is that our body realizes that we need some very important things such as sodium and potassium. Let's take it back. So this reabsorption that happens, my dear children, it happens in the PCT, and this is known as reabsorb. Basically, what do you call it as? Selective reabsorption. You call this as selective reabsorption because you're selectively taking in some very important things. You do not take everything up. Selectively take things up, whichever it's needed. So this is also correct. This can happen in PCT. Now, loop of Henle, which is there, it is responsible for concentrating the urine. Concentrating the urine as in whatever amount of H2O can be taken back will be taken back, so that the urine will not be having too much amount of water. We do not want to lose water, my dear children. So basically the role of Henle's loop is to concentrate the urine. So loop of Henle is responsible for urine concentration. What are we left with? We are left with DCT. So my dear children, until now we are taking back a lot of things out there. Now suppose our body feels that, oh my god, these ions are very much in our body, or some toxic substances are very much in our body, we need to give it back to the nephron. And where does that happen? It happens in the DCT. In the DCT, reabsorption of glucose does not happen. Rather, if glucose is way too much, it is given back to the DCT. This is known as tubular secretion. It is known as tubular secretion. So reabsorption does not take place over here, which is why I'd say this particular statement would be entirely wrong. Okay, so choose the mismatched pair. This is the mismatched pair. Okay, 14th question. Select the correct statement regarding the specific disorder of muscular or skeletal system. Okay, you have to find the correct one. So whenever you have to find the correct one, underline that. Muscular dystrophy, age-related shortening of muscles. Does everyone have it because we are aging? No, Baba. It's a genetic disorder where you have weakening of the muscles. There is no shortening of the muscles and it is not at all age-related. This particular thing would be definitely wrong. Osteoporosis, decrease in bone mass and higher chances of fractures with advancing age. Yes, there's a loss of calcium from the bone, which is why there is a decrease in bone mass and it becomes brittle. There are chances of easy fracture also. So this is correct. This particular statement is going to be correct. Myasthenia gravis, autoimmune disorder which inhibits sliding of myosin filaments. Myosin filaments are there in the muscles. Now listen to me carefully. It's an autoimmune disorder where it affects the neuromuscular junction. You know, between a neuron and a muscle, the junction that you have is known as neuromuscular junction. Now in this junction, my dear children, you have some receptors which are known as acetylcholine receptors. Acetylcholine, you know, it's a neurotransmitter. These receptors are there. Now suppose someone is blocking this receptor such that the acetylcholine does not come and bind. Yes, that is the problem that is happening over here in myasthenia gravis. Okay, so there is no problem in inhibiting the sliding of myosin filaments, no, nothing like that. So this would be wrong. Now gout, inflammation of joints due to extra deposition of calcium. Yes, it happens in joints, but it's not because of calcium. It happens because lots of uric acid crystals are being accumulating out there, which is why there's a pain and things like that. So obviously this particular statement is also going to be wrong. So what is the correct statement that we are having? It will be B, small b. Okay. Yes. So over here I have written everything. Myasthenia gravis is an autoimmune disorder. It's an autoimmune disorder in which antibodies attack the receptors for acetylcholine at the neurotransmitter junction, preventing proper nerve to muscle communication out there. Got it? The system that transmits impulse from the CNS, central nervous system, to the involuntary organs and the smooth muscles. Remember, involuntary as in it's not under our control. And smooth muscles are also involuntary muscles out there. For example, muscles which are there over here. So food, you take it in, that's voluntary. But everything that happens inside, do you think it's under our control? Can you say that, what do you call, esophagus, please do not do peristalsis. Can I do that? No, Baba. You cannot, because all of them is involuntary. And all the involuntary functions are done by our autonomic neural system. And under the autonomic neural system, you have sympathetic and parasympathetic neural system. So if at all you have a broad thing to be asked, then it is autonomic neural system which is taking the control. Somatic neural system controls all the voluntary functions which are there. Got it? Yes. And with that, we end all the MCQ questions, and now we enter into the next thing, that is what? Basically the fill in the blanks question. So let's see. In dicot root, the endodermal cells have a deposition of water impermeable waxy material, suberin, in the form of, in the endodermal cells, my dear children. So if at all you see the endodermal cells, this is cell wall. Suppose this is cell wall. They will be having some strips like this in between. And this strip is actually made up of suberin. What is this strip? This strip is actually made up of suberin, and this is known as Casparian strip. This is known as Casparian strip. You do not want anything to move through the cell wall or the intercellular region. That is why you have Casparian strip. Anything that has to move will be moving through the, what do you call, the cytoplasm. Okay, and it will be selectively moving. Not everything can move. You get it? So in dicot root, the endodermal cells have a deposition of water impermeable waxy material, suberin, in the form of what? Casparian. Here it is written Caspian. It is Casparian strip. Okay, so this would be your 16th question's answer. Now, the process when dedifferentiated cells again lose the ability to divide and get mature is known as. So listen to me, it's very simple. Okay, so first thing is that a cell is differentiated. That means it is mature. It knows what it has to do. It knows its function and everything. Suppose when I say it is dedifferentiated, I try to mean that basically as of now, okay, so when I say it's mature and it is differentiated, I also mean to say that it is not able to divide. It will not be able to divide. It is in differentiated stage. Now, when it comes back to the immature form, that is, it is now able to divide, you call the stage as dedifferentiated stage. Dedifferentiated stage. It has the ability to divide now. Again, when they come back to a stage where, my dear children, they have lost the ability to divide, you will be calling this as redifferentiated stage. So whenever something is differentiated, you mean that they are mature, they are not able to divide. They have lost the property to divide. So over here, the process when dedifferentiated cells again lose the ability to divide and get mature is known as redifferentiation. So redifferentiation is given right over here. Your answer for the 17th question is this. Now, the respiratory rhythm is maintained by the respiratory center in dash region of the brain. I told you, the respiratory rhythm and your heartbeat and everything, my dear children, is controlled by medulla oblongata. Okay, in the medulla region, of the brain. That is how you need to answer. Now, dialysis fluid contains all the constituents as in the plasma except. So listen, dialysis fluid is, dialysis happens for the patients whose kidney is not working properly, right, who has some trouble with the kidney. Now, over here, the role of kidney is to remove the nitrogenous base such as urea, uric acid and things like that. Oh, okay. So when someone is doing dialysis, what is going to happen? They have a chamber being made like this and there will be fluid. And they will be having some structures like this through which the blood is going to pass. The blood is going to pass to the structure. Now suppose over here in the fluid which is there, that is dialyzing fluid, if at all I have too much amount of nitrogenous waste, if at all there is too much of nitrogenous waste, what is going to happen? From this, it will be getting inside my blood, and that I do not want. I actually want to remove from the blood towards the dialyzing fluid. So which is why we will make sure that there is no nitrogenous waste over here. Why? Because in that particular condition, whatever nitrogenous waste is over here inside the blood will be moving out from a region of higher concentration to a region of lower concentration. So that is the logic. So the dialysis fluid contains all the constituent as in the plasma except what? Except the nitrogenous waste out there. Got it? Yes, ma'am. Now, dash, 20th one. Dash helps in maintaining the normal rhythms of sleep-wake cycle, body temperature in our body. What do you think is responsible for maintaining the normal rhythms of sleep-wake cycle? Body temperature? It is something that is known as melatonin. It is known as melatonin. Now, this is super important. When we are using mobile phones and all, it leads to release of melatonin, which is why we are not able to sleep really well, which is why we say that please put your phones away. Yes? So with that, we have completed all the 20 questions of part A. Now, if at all you want, you can take a chutku break. Okay, chutku break and then be back, as in just go through the questions and be back. Okay, so I hope you are done with your chutku break out there, and now we are back again to solve part B and part C questions, that is the 2-marker and 3-marker question. So our next break will be post after that only. Until then, it's just you and me, and we are going to ace it. Yes? And the first question that we have from the 2-marker session is this. That is, what are the advantages of giving scientific names to organisms? Why do you think organisms have some scientific names out there? The reason is that, for example, when you talk about cockroach, you know what do you call cockroach as? The scientific name of cockroach, American cockroach to be specific, is Periplaneta americana. Now, such a fancy name for a cockroach, right? Why? Because if at all you go to North India, at some places, okay, at some places they call the cockroach as tilchatta. Okay, now if you go to South India, I am a Malayali, so I know that in Malayalam you call it pata. Now you have different names for an organism, and with this information, do you think there will be any sort of universality? Universality, exactly. When you go to different places and say, oh, there was a pata there, or there was tilchatta there, do you think they can understand it, or do you think the scientific study of this organism can be done? You know, this is a organism that causes lots of trouble. It also passes lots of diseases, right? So obviously we need to have something which is universally correct. at every point of this particular universe, it will be having the same name itself. So that is the reason that we give scientific names. Now Periplaneta americana, for everyone it will be what? It will be a cockroach. That is the American cockroach out there. You get it? So that is the use. Second thing, uniqueness. For example, today I call a cow a puchu. Okay, today I'm calling cow as puchu, and tomorrow I'm calling someone else is calling what do you call a dog as puchu. You get it? So there are two organisms with the same name, and that is not, if at all you want to study about them, it's not okay to have the same name for multiple organisms. So for maintaining the uniqueness, and remember, scientific names are not repeated. So scientific names are unique names given to certain organisms out there. So for maintaining universality and also uniqueness, you have basically, you are giving scientific names to the organism. Scientific names are accepted worldwide, eliminating confusion caused by local or the common names out there. Uniqueness, each organism has a unique name, avoiding duplication and also misidentification. Now all of this I explained it to you in a jocular way, but that is not exactly how it works in the terms of research as well as development, which is why there is too much amount of specificity that is needed. Okay, now next question. Name the animals exhibiting the below given characters. Organism with sucking and circular mouth. So circular mouth can also be known as cyclostome. Cyclostome means circular and stoma basically means mouth. So they have cyclostomes, my dear children. They have a circular mouth, and you know what? They do not have jaw. We have jaw. This is our lower jaw. There is an upper jaw onto which our teeth is there, right? But they are organisms that do not have jaw. Okay, so which is why I call them as agnatha. What do I call them? Agnatha, the ones without jaw. So ma'am, they don't have anything like that to crunch up things. They do have. And my dear children, it looks like this. So their mouth is like this, and they have an arrangement like this. And my dear children, with that arrangement, they can suck into some places and have whatever they want. So organism with sucking as well as circular mouth is known as agnathans, or you particularly call them as cyclostomes. And the example for them is petromyzin, lamprey, and also myxin, that is hagfish. Okay, petromyzin, PM, Prime Minister. Petromyzin as well as myxin. Cool? You also call petromyzin as what do you call lampreys, and myxin as hagfish. So cyclostome, I hope it's done. Okay. Now they are vertebrates, okay? They are coming under vertebrata. Now going ahead, organisms in which notochord is present only in the larval tail. Organisms in which notochord is present only in the larval tail, you call these organisms as urochordates. Okay, what do you call them? Urochordates. So basically, I'll tell you again, when you talk about vertebrate, when you talk about what do you call what is that name? Just a second, okay? Okay, yeah, when you call organisms that they are under chordata group, so basically it means that they have a notochord. Now these organisms are of three different types. One organism is urochordata, but the notochord is there, but only in the larval tail. So when it is a larva, and the tail of the larva would be having this particular notochord. So those organisms are known as urochordates, and that is your question. The organisms which have notochord present only in the larval tail. Now going ahead, you have cephalochordata. Cephalo means head. Okay, cephalochordata means those are organisms which have the notochord from the head to the toe region, and my dear children, okay, they have the notochord throughout their lifetime, not just in the larva, they have it throughout their lifetime. Now talking about the next group of organisms, these are vertebrata. Vertebratas are one where the notochord is actually replaced by what? The vertebral column. And under that comes agnathans and also gnathostomata. So under gnathostomata, the organisms that have jaw will be coming up, such as all your fishes, amphibians, reptiles, and all of this thing. And agnatha, as I told you, cyclostomes will be up there. Got it? So your question is, organism in which notochord is present only in the larval tail is basically a urochordate, that is Acidia. These organisms belong to the phylum Chordata. So as I told, Chordata has three groups: urochordata, cephalochordata, and also vertebrata. Okay? Done. Now, next thing, frogs can live both on land and in fresh water. Write a note on the respiratory system found in frog to survive in both of the habitats. So they are amphibians. Amphibians means the organisms that can live both on land as well as water. Now, when they live in water, obviously at that particular condition, your lungs is not something that could be supporting you, right? Yes. So what do you need for respiration? Something else. And what is that something else? In the case of water habitats or aquatic habitats, it is basically the skin. So frog basically respires through the skin, and this type of respiration, which is known as what? Cutaneous respiration. What do you call this? Cutaneous respiration. Now when they are, okay, just read the next line also. Dissolved oxygen in the water is exchanged through the skin by diffusion. Okay, so in the water you have dissolved oxygen, and the amount of oxygen would be very, very less. Okay, it's not like the atmospheric oxygen that we have. In the water, that dissolved oxygen is pretty less, and my dear children, it is exchanged through mere diffusion with the help of the skin in between. That's all. Right? Now on land things are going to be different. On land, the first thing that you can possibly remember is what? Having a lungs. So yes, lungs is going to play a major role out there. Then you have your skin also. Not your skin, it's frog skin, yeah. So frog skin also helps in respiration out there. And apart from that, there is buccal cavity, which is also going to help in respiration. So what do they do is that they close up their mouth, okay, and the floor of the tongue, which is there, my dear children, or the floor of the buccal cavity, which is there, it has been pulled up and down. By closing there, the nostrils are going to be open, and basically they are going to close their mouth, and they are going to flap up their buccal cavity, the lower part of the buccal cavity, which is how they are going to trap in the thing out there, that is the air out there. That is how respiration happens to the buccal cavity. Now I do not know how far have you understood this, but it was there in the NCERT, so I thought of anyway explaining this. Anyway, just understand it is buccal cavity also, and with this much amount of explanation, obviously you will not forget that there is something like buccal cavity also involved in respiration, right? So that is a good part of the story, even if you have not understood the other part. Okay, so buccal cavity, skin and lungs are the respiratory organs during aestivation and hibernation. What do you mean by that? So organisms, they go into a stage where the metabolic rate is very, very less, and that is known as aestivation and hibernation, right? And at this particular point of time also, their metabolic rate is less, but that does not mean they won't be breathing, right? They would be breathing, and that would be happening through skin. Okay, so this is what there is there in your NCERT. I hope you have got it. Now just give me a second, okay? Okay. Now, the next question that we have is 24th question. Identify the cellular structures with the help of the following features. So, features are given over here. You have to find out what is that cellular apparatus out there. So, these are the sites of aerobic respiration. So where does respiration begin from? Respiration begins from your cytoplasm. What happens in the cytoplasm? Glucose is broken down to pyruvate. We all know that, right? Now, if at all you're talking specifically about aerobic respiration. Aerobic respiration means it has to happen in the presence of oxygen. Now, the pyruvate that has formed, it has two things to do. Either it can undergo anaerobic respiration, that means in the absence of oxygen, respiration will be happening. That is going to happen in the cytoplasm itself. Now, if at all you want aerobic respiration, that is in the presence of oxygen, it has to enter the mitochondria. In the mitochondria, it will convert into acetyl-CoA, and then the, all the cycles are going to happen. The Krebs cycle, DCA cycle, everything will be happening up there. Got it? Okay. The Krebs cycle, that is a TCA cycle. Apart from that, the DC cycle, at the end you get the energy format. That is how it usually goes. But anyway, what is the place where aerobic respiration is going to happen? Exclusively in the mitochondria. If you're asked about anaerobic respiration, then it would have been cytoplasm. Got it? Yes. Now, it destroys foreign substances. So there is only one cell organelle in our body which is responsible for all the destruction. And it's not like it's destructing because it wants to. It's very impulsive. It's not like that. Basically, it's its role. They are known as the suicide bags of the cell. Why? Because they are ready to give their lives for the cell, my dear children. So anytime the cell is having some problem, my dear children, they are, my dear children, having so many enzymes, they can release those enzymes and kill the whole cell. If at all they think that the cell is under danger. Similarly, any foreign substances which comes in, it has the power to destroy those things also with the help of the enzymes that they can secrete. So the name of this particular organelle is what? Lysosome. What do you call this? Lysosome. Fine. Next thing, it gives turgidity to the plant cell. So turgidity, my dear children, if at all you see, the plant cell if at all you have ever noticed the plant cell, they have a very big vacuole. They have a very big vacuole, and this vacuole which is there is the one that provides turgidity, the proper structure to the entire plant cell. So your only answer for this question is going to be a vacuole. Now, it initiates and regulates cell division. What do you think it is going to help in the process of cell division? So cell division is a process where basically, yes, organelle duplication and all of this thing happen. Apart from that, my dear children, the chromosomes which are there, they also need to be pulled about such that they can be spread equally. Right? Now for that, what do you need? You need one very important thing which is known as centrosome. Okay, and these centrosome, my dear children, will be having centrioles, which is responsible for the formation of spindle fibers, which are going to separate your chromosomes in two particular directions. They are like the ropes. Have you ever played tug of the war? So both of them pull apart, right? Similarly over here also, it's like that. So it's a centrosome, and the centrosome, what do you have? You have centrioles, which basically helps in the formation of spindle fibers, and yes, it definitely helps in the process of cell division as well. Done? Done. Okay, next, why is interphase of the cell cycle called the resting phase? Okay, so you know what, if at all you talk about a cell cycle, almost 95% of the time the cell is in its interphase stage, and the 5% time is actually the time needed for the mitosis or the cell division to actually happen. Okay, so ma'am, why 95% is this particular cell in the interphase stage? Because, my dear children, also why do you call it resting phase? I'll say it's, so when you rest, my dear children, okay, generally when you have a resting day, we be more productive, right? We have more energy to do a lot of things. So you can imagine like that only. Interphase which is there is divided into multiple phases. You have G1 phase, you have S phase, and you have G2 phase. And in all of these places, some things are happening. And what are these things? These things are super important for the actual division to happen. So it's like half, not half, more than half of the work, 95% of work is done, and only 5% work will be left by for the actual cell division to happen. You get it? So what is happening in the G1 phase? In the G1 phase, you know, when a cell is going to divide, okay, obviously this cell will make sure that all the organelles, whichever is there, my dear children, it has to distribute these organelles in the same way to both the cells out there which are going to come out of it, right? So over here, organelles which are there, they are going to be duplicated. Whichever major organelles are there, they are going to be duplicated. What about the S phase? S phase is also known as synthesis phase, and what exactly happens over here? The DNA which is there, it is going to be replicated. Again, organelles, they are duplicated, done. Now DNA also you have to do the same way, only divide equally. So which is why the DNA is going to be replicated in this phase. What happens in the G2 phase? G2 phase is where the other organelles which were left behind, they duplicate. Apart from that, there are some very important proteins needed for the cell division process. Those are also being made up during this particular phase. So basically, when I say that this whole thing is interphase, is resting phase, it's not like it's actually resting, it's preparing itself for the actual cell division which is going to happen. Okay, so interphase is called the resting phase because there is absence of cell division. There is no cell division happening up over here, right? Do you see any cell division? No. And no active mitosis or meiosis occurs during this phase. Now, metabolic activity continues. Okay, that is also a reason that the cell grows, replicates DNA, synthesizes proteins, organelles, preparing for the next division. So metabolic activity is continuing, but there is no cell division, which is why you call interphase as what? Resting phase. Got it? Yes. Next question. Abscisic acid is also known as stress hormone. Give reasons. So, okay. Why do you call this as a stress hormone? So it's like the plant which is there. Okay, whenever plant is under stress, for example, it does not have too much amount of water. It does not have too much amount of water. Now, plant, do you think it can grow very easily if there is no too much amount of water? Suppose there is no too much amount of carbon dioxide. It cannot do photosynthesis. How will it make its own food? All of it is a very big problem. Now, under this particular condition, do you think the plant can easily survive if at all there is no one to help? No. Obviously, it needs someone to help. For example, in this particular case, when the plant does not have enough amount of water in itself, what is the plant going to do? It will make sure that at least the stomata which are there, they are closed. Because if the stomata remains open, whatever water is there, that will also get lost, because sun baba is there, sun will cause evaporation to happen. You get it? Which is why the stomata has to be closed. Who gives the information that yes, the stomata has to be closed? It's the abscisic acid. So which is why I'll call this as a stress hormone. So during it's not like it's causing stress. No, when stress is there, that's when the plant is going to produce abscisic acid to basically be safe from the stress. Okay, apart from that, suppose I have a seed. Okay, I have a seed and I am putting it in an area where there is no water, there is no nutrient, nothing. Now at this particular stage, do you think the seed should be germinating and give rise to a whole new plant? No, Baba, that is not possible at all. At this particular stage, the seed should stay dormant. Why dormant? Because it does not have any other nutrient medium or the water medium to grow. So at this particular time, it has to shift to what? Dormancy stage, where the metabolic rate is going to be less. Who gives the signal for that? Again, abscisic acid. Got it? Yes. So basically, abscisic acid, ABA, is also known as stress hormone because it induces dormancy. It helps plants survive unfavorable condition by inhibiting growth and seed germination. Okay, as I said, during unfavorable condition, it does that. Now, it regulates stomatal closure. During drought or water stress, ABA causes stomata to close, reducing water loss. These functions help the plant cope up with the environmental stress like drought, cold, as well as salinity. Salinity means too much amount of soil in the, too much amount of salt in the soil. Okay, at that particular point of time also, you do not want the plant to grow. So that is the reason why you call this as the stress hormone. Next, define tidal volume and residual volume. It's very simple. Tidal volume means the volume of air that I am inspiring and also expiring during what? A normal respiration. And do you know how much approximately it is? It is approximately 500 ml. Now in 1 minute, how many times do you breathe? Usually 15 to 16 times. And if at all you do that calculation, you will be coming into, so whatever number of times we breathe into 500 ml, my dear children, you will be getting these numbers. So basically this is what the approximately tidal volume of a healthy person per minute. Okay, per minute data is this. As I told you, per minute you might breathe 15 to 16 times or 12 to 16 times. In whatever range, if at all you calculate it with 500 ml, you will be able to understand what is your whole thing per minute. Got it? Yes. Now, so tidal volume is what? Volume of air inspired and volume of air expired, approximately 500 ml. In healthy individuals, it becomes 6,000 to 8,000 milliliter per minute. I'm talking about per minute here. Fine, done. Now residual volume. Residual volume is that, for example, my dear children, all of you together, okay? When I count to 3, everyone do expiration. Breathe out, okay? 1, 2, 3. Okay, I did that. Now do you think all the air in your lungs have been moved out? No, there will be some amount of air still living back, and this is known as the residual volume. The volume of air remaining in the lungs even after a forcible expiration. You're doing maximum of what you can, and then also there will be something left. Otherwise, your lungs and everything will collapse, my dear children. So the average, this average is 1,100 to 1,200 milliliter. 1,100 to 1,200 milliliter. Will you forget this? This is what the residual volume. Okay, done. Tidal volume is 500 and per minute it is 6,000 to 8,000. Cool. Next. Okay, so guys, we have done the second, what do we call, the 2 marker things also. Now we are entering into the 3 marker thing, and yes, it is hopefully going to be good. By the way, if at all you are liking the video and yeah, you're not getting bored, if at all it's like that, do not forget to hit the subscribe button and also share it with your friends, and do not forget to let me know in the comment section also. Also, if you have doubts, please let me know in the comment section. I'll take it on priority. Yes. Now, write the stored food present in the different classes of algae. Okay. So, these are the 3 different classes of algae. Chlorophyceae as in green algae. Phyophyceae as in brown algae. Rhodophyceae as in red algae. Okay. Done. Okay. Now, chlorophyceae, it stores protein and starch. So, you are being asked what is the stored food present. So, it is mostly the proteins as well as the starch. It is stored, the food is stored in the form of starch. And in some algae, it may store food in the form of oil droplets also. So, basically 3 things. Okay. It can store things in the form of starch. In some of them, they store in the form of oil droplets, and mostly they store protein. So that's not a form of things being stored. It's like a thing. So it's just like what do you call? I'm forgetting names. I don't know why. Okay, anyways, let's just leave it, otherwise it will be taking too much of your time. So in chlorophyceae, the food is stored in the form of starch, and in some it is stored in the form of oil droplets. Just remember that. Done. Okay, next thing. Phyophyceae, that is the brown algae. Here the food is stored in 2 formats, actually in the form of complex carbohydrates out there. And my dear children, the name of these complex carbohydrates which is there is mannitol and laminarin. Mannitol and laminarin. You can read it in what do you call? You can remember it in different ways. Lead manager or mother-in-law, whatever you want to remember, you can remember. But please remember laminarin and mannitol. This thing has usually come up, right? So that is why I am asking you to remember this. So in Phyophyceae, that is the brown algae, it is stored in the form of complex carbohydrates and usually laminarin as well as mannitol. Okay. Next thing, Rhodophyceae, that is the red algae. Here the food is stored in the form of fluoridine starch. Remember this name, fluoridine starch, which is very similar to amylopectin and glycogen in structure. So fluoridine starch which is there, it is similar to amylopectin and glycogen. Remember that also, amylopectin and glycogen. It is so fluoridine starch which is there, it is resembling or very similar to these structures that you see over here. Done. So once more let's revise. In Chlorophyceae, green algae, it's usually in the form of starch only. Apart from that, my dear children, also in the form of oil droplets in some. Okay, in brown algae, laminarin and mannitol. Actually in the form of complex carbohydrates, examples for that is laminarin and mannitol. Okay, in red algae, it is fluoridine starch, and this fluoridine starch which is there, it has high resemblance to two things, amylopectin and also glycogen. Done. Okay. Now, and mannitol, my dear children, it is sugar with alcohol. Okay, that is why it is all the term when it comes, it is sugar with alcohol. So yes. Now, the tissue between the upper and the lower epidermis of a leaf is known as mesophyll. So my dear children, if at all you consider a leaf, let's consider a leaf, okay? And this leaf I'm cutting like this. Fine. So, and I'm looking it like this. So I have cut the leaf. Okay, suppose I have cut the leaf and now I am looking in this direction. So what is there over here in this direction? So I would be able to spot the upper epidermis. Apart from that, I will be having the lower epidermis with guard cells and everything. And in between that, whatever you have, you call this as what? Mesophyll. That is the statement being put down over here. Okay, so tissue between upper and lower epidermis of a leaf is known as mesophyll. Fine. Now, write the type of cells found in this tissue in dicot leaf. So in the case of dicot leaf, the things are going to, there are two type of cells out there. Okay, one is palisade mesophyll or palisade parenchyma, which is like this. They run very parallel to each other. They are arranged very parallel to each other, and my dear children, they are elongated cells. Okay, they are elongated cells. So palisade parenchyma, they are elongated cells arranged parallel to each other, and my dear children, one very important thing, they will be having multiple amount of chloroplast. So when you say that the leaf is responsible for photosynthesis, this particular area is the thing that helps most in photosynthesis. What is it? The palisade parenchyma layer or the palisade mesophyll layer. Okay. Now apart from that, they also have one more thing, that is spongy mesophyll. And in spongy mesophyll, the cells are like this. The cells would be like this. So what do you understand from that? Basically, it is either oval or round. It is either oval or round. There it was elongated, and also it is loosely arranged. You can see much space up there. Here it was parallelly stacked very close to each other, right? Here it is loosely arranged, and here also, my dear children, you would be having some chloroplast. It's not like there is no chloroplast. Here also some chloroplast will definitely be there. And my dear children, why do you think there is space in between? Because that facilitates gaseous exchange. It is going to help in gaseous exchange. So in dicot leaf, you have palisade parenchyma and you also have spongy parenchyma. Okay. Now next question is there. Mention any two anatomical difference between dicot leaf and a monocot leaf. So the first thing that is very easy to remember in a dicot is like, I used to teach children like this. Doctor. Okay. Doctor. Starts with D, ends with R, right? So here in the case of dicot leaf, if at all you have seen, their leaf is like this. Normally, how do we make leaf? We make the leaf like this, right? This is how it goes. And this is known as reticulate venation. Okay, this system of arrangement of veins are known as reticulate venation. So dicot read reticulate venation, doctor. Okay, and what about monocot leaf? Monocot leaf, my dear children, will be showing parallel venation. So MP, Minister of Parliament. So over here, if at all you see their leaf, it is going to be like this. It is going to be parallelly arranged like this. Okay, so where am I? Okay, dicot leaf shows reticulate venation, network like vein arrangement, and monocot leaf show parallel venation, that is member of parliament. Done. Apart from that, dicot leaf, you saw that the mesophyll is differentiated into palisade as well as spongy, whereas when you talk about monocot leaf, my dear children, the mesophyll is not differentiated into palisade and spongy layers. It's just plain mesophyll. So you do not see any differentiation of multiple different type of cells out there in the case of monocot leaf. Done. That's all. Fine. Next question. Draw and label the parts of an organelle responsible for trapping light energy, essential for photosynthesis in plants. So what is that thing that traps light energy? Baba, it's chloroplast, right? It's chloroplast. Inside chloroplast, what is there to trap? It is chlorophyll pigment that is responsible for this. And now if you're asked to make the diagram of chloroplast, how do you make it? So basically you can do something like this. Over here, there are two membranes in the case of chloroplast, the outer membrane and the inner membrane, right? Apart from that, you have thylakoid. They are coin-like structures, and the thylakoid which is there, so please mention it properly. It is thylakoid, and thylakoid are arranged on the form of stack, and that is known as grana. Okay, now you will be having multiple stack like this, and these stacks are joined together with the help of something that is known as stroma lamellae. Okay, what do you call it? Stroma lamellae. And my dear children, this thing happens. Okay, this is continued out there. Now over here, this extra region that you see is known as stroma. Do not get confused between stroma lamellae and stroma. Stroma lamellae is this, and stroma means the extra space that you see over here. This is generally what you have to talk, and please mention that there are chlorophyll pigments also. Please mention the chlorophyll pigments as well. Okay. So yes, the inner membrane and outer membrane is there. Apart from that, you have some fluid filled spaces out there. The extra thing, whichever would be there, please mention those also. Lipid globules, starch grains, and granum is also being mentioned, right? So yes, whatever extra you see in this picture, you can mention that also. Done. Next thing. Plant cell undergoes cytokinesis by different mechanism rather than in animal cell. Substantiate. I'll talk about animal cell first because it is very easy to discuss about that. Okay. So animal cell, my dear children, if at all we talk about an animal cell which has underwent division. So it has number of chromosomes over here, number of chromosomes over here. So this is the nucleus and everything, right? How does it undergo cell division? Over here, from here to here, there is a movement inwards. Okay. So basically you could see something like this happening. There is an inward movement, and when this inward movement happens, my dear children, at the end you will get two cells. You will get two separated cells. Why is this very easy to happen in the case of animal cells? Because animal cells do not have cell wall. Do not have cell wall. So basically this type of cell division is known as cell furrow formation. Cell furrow formation through mediated cell division. Okay, here cell furrow is being made. And why is it very easy for happening in the case of animal cell? Because they do not have cell wall, so there is no one to stop it. But now if at all you compare it with a plant cell, do you think plant cell can undergo this? No. Plant cell has a very tough cell wall made up of cellulose, pectin, lot of things out there. Do you think it can take things in? So which is why we have to start from inside and go outside. Oh, how exactly does it happen? So you might have heard about Golgi apparatus. Okay, in the case of plants you call them dictyosomes as well, because that is a more advanced form of Golgi apparatus, as a specialized form of Golgi apparatus actually. And they release one very important, they release so many vesicles. Okay, they release so many vesicles filled with a lot of things. These vesicles which are there, they join together up over here, suppose, and basically it will make a division that goes through like this, through the cell like this. This is known as a cell plate. And the division happens through this cell plate itself. So there's a cell plate formation that happens. A cell plate forms at the center of the dividing cell. Vesicle fusion. Golgi derived vesicles containing the cell wall material. Okay, it has the cell wall material. It fuses at the equatorial plane, as in the middle plane. Cell wall formation starts in the center. So now the cell wall, since it has the vesicles with all the materials needed for cell wall formation, slowly a cell wall formation happens over here, and my dear children, that is how it is going to get divided. So cell wall formation starts in the center of the cell and grows outward to meet the existing lateral wall. So these are the lateral walls out there, it goes out there. And this is known as cell plate formation. Okay, cell plate formation. Now next thing, cytokinesis in the case of animal cells. There is furrow being formed. So cleavage furrow formation. Appearance of a furrow in the plasma membrane, because they do not have any cell wall. The furrow gradually deepens and ultimately joins in the center, dividing the cytoplasm into two. Separation. The membrane eventually splits, and now you have two cells out there. Thus plant cells form a cell plate, while animal cells undergo cleavage with the help of cell furrow. That is how things happen. Easy, right? Easy. Okay. Next thing, write the characteristics of plant growth regulators, PGRs. What do you mean by plant growth regulators? Basically, you are talking about the hormones which are playing a very important role over here. There are two types of hormones, okay, or plant growth regulators. One that helps in plant growth, one that will be not allowing the plant growth to happen. Or basically, I can say one that inhibits and one that promotes. Okay. Now, one that promotes are of three different types actually: auxin, gibberellin and cytokinin. Auxin, which is there, is responsible for cell elongation. Suppose your cell was like this, it will elongate the cell to be like this. Now obviously isn't it promoting the growth of the plant? Obviously, when the cell which are like this, they are growing in size, obviously the plant is also growing. So it also promotes cell elongation. Apart from that, root initiation, it helps in the formation of root. And apart from that, apical dominance. What do you mean by apical dominance? Apical dominance means, over here my dear children, when you have branches, you call them lateral development. Okay, and when you say that the plant is just growing like this, it's not allowing lateral growth, rather than it grows like this. This is your apex. And when I am saying apically it is dominant, that means that over here it is promoting this type of growth rather than the lateral growth out there. So auxin is responsible for apical dominance. Now if at all, there is a question that is usually been, I just came into my head. If a gardener is there and he wants to make the plants bushier, okay, too many branches and things like that, first of all he would have to cut the tip of the branch. Why? Because at the tip you have auxin that promotes what? Apical dominance. Then only, if at all that guy has gone, then only there will be lateral growth that would be seen. Okay, so auxin is responsible for promoting cell elongation, root initiation and also apical dominance. Now gibberellin, G for gibberellin and G for germination. G for gibberellin and G for germination, my dear children. It helps in the germination of the seeds out there. Apart from that, it also helps in flowering. So the flowering blossoms out there, it's because of gibberellin. Apart from that, they also help in stem elongation. So both of them has kind of okay, so both of them are involved in elongation. One in cell elongation, here it is stem elongation, and yes, seed germination and also flowering is there by gibberellin. Now cytokinin, the name itself is super easy to understand. Cyto means cell, kinin means division. So the one that promotes cell division is known as cytokinin. How do you think cytokinin or cell division can help in the plant growth? If the cells are dividing, then only plant is going to grow, right? Otherwise, do you think plant will grow? No. So it promotes cell division, it delays leaf senescence and helps in shoot growth. It helps in shoot growth, and delays senescence means delays aging of the leaf. That is what you mean by that. Okay, so basically all of them has growth promoting features: auxin, gibberellin as well as cytokinin. Now you have growth inhibiting regulators out there. You have abscisic acid. Now you think that okay, it is inhibiting growth and it is a bad boy. No, we have studied about it that abscisic acid does not allow growth, because the plant, if at all it tries to grow, it might die. There are no favorable conditions out there, and at this particular point of time, there you have to be the bad cop. So just like your parents, sometimes we feel that they are not allowing us anything. What is this life and blah 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 blah. We go on thinking like that, but sometimes we have to realize it's for something good also. Okay, you realize it, don't worry. Okay, so when you will grow up, if at all, and most of the times our parents are really great, right? And we do realize that whatever they said comes out to be sometimes true. Most of the time it's true itself. So yes, that realization will come slowly. Okay, now abscisic acid induces dormancy as in it does the opposite of what it was doing. Gibberellin was doing seed germination, but it does the opposite. It induces dormancy, closes the stomata during drought and inhibits seed germination. So all of it is a negative factor, but actually it's for good. Now ethylene, which is there, that is next negative growth factor out there, or inhibiting growth factor out there. What does it do? Basically it promotes fruit ripening. Okay, it promotes fruit ripening, leaf abscission and inhibits stem elongation as well. So opposite of what cytokinin was doing, right? It was delaying leaf senescence. Here it is doing the opposite. But the major function of ethylene is to do fruit ripening. So ethylene is a gaseous hormone. Remember, it is a gaseous hormone, the only gaseous hormone that you have. And you might have seen that whenever a fruit is ripened, we keep the other fruits also to ripen just beside them. Why? Because the fruit which has already ripened release these gases, influencing the other fruits also to ripen. That is how things work. Okay, so it's a fruit ripening thing, and it promotes leaf senescence and also inhibits stem elongation. Done. Okay. Next thing, draw a neatly labeled diagram of a nephron. Just a second, okay? Okay. 2 more questions. Then we are done with part 3. Okay. Neatly labeled diagram of nephron, how do you make it? Very simple. So listen to me carefully. First thing that you have is a bowl. Bowl of what? Noodles. Yes, you will have a bowl of noodles. I didn't have to cover it up. Just a second. Yes. So over here you have a bowl of noodles, and you are going to spill some noodles out right like this. These noodles that you are going to spill. Okay. So first of all, what is this bowl? It is known as Bowman's capsule. Now, they are tubules, and these tubules are very proximally located to the bowel. So I'll call this as proximal convoluted. Why convoluted? Because they are like twisted and turned tubules. Okay, now what happens? Basically, there is something like this being found. This is your loop of Henle. What do you call it? Loop of Henle. Loop of Henle will be large over here, get small over there. This side of loop is known as descending loop of Henle, and this side would be known as ascending loop of Henle, which is tiny first and then it grows to big. Now over here again you have some convoluted tubules, but they are actually far from the Bowman's capsule, which is why you call them distal convoluted tubule. Okay, and they open into a duct like this, which you call as collecting duct. So multiple nephrons will be opening into this particular duct. Okay, got it. Now what? Now listen to me. So basically, you know, renal artery is there. That is, renal artery provides oxygen as well as other nutrients to basically the kidneys out there. Now the renal artery has to divide into multiple arterioles. Why? Because multiple nephrons are there, and to each of them you have to provide whatever has to be removed out and also whatever has to be taken in. So basically the artery which is there, renal artery which is there, is divided into multiple arterioles, and you call them as afferent arteriole. Afferent arteriole. Now the afferent arteriole is coming over here and basically making a bunch of capillaries out there. You call this as glomerulus, and then it opens into next artery. So it's like, usually capillaries come between arteries and veins, but here it's like a bigger artery, that is afferent arteriole came, it made branches or basically capillaries, and now it's opening into a smaller arteriole that is known as efferent arteriole. It's efferent, E for exit. So this is taking an R and this is E arteriole. And here exchange of things is going to happen. That is known as glomerular filtration. I have taught you that, right? Now listen to me very carefully. This particular thing which is there, it is going to run around the your entire structure out there. Why it is running around the entire structure out there? Basically, so I have not made a really good picture, but yes, basically it's running around the entire structure because, you know, it has taken up things out there, but you know what? Certain things would be given back to the blood. So over here filtration happened. Everything which was small and everything passed down, but it's not like everything was not useful for us. It is still useful for us, which is why it is going to get reabsorbed by our blood vessels. Okay, and also the urine which is there, it has to be made concentrated. The amount of water and everything which is there, it has to be removed also, which is why you need a lot of blood vessels around it, and you call it as vasa recta. All of these blood vessels it will be covering that portion also. Okay, all of this blood vessels will be opening into what? Into a vein that will be taking away whatever is not needed. Okay, fine. So it actually looks like this. So you have to cover the other part also. So you can make it like this as well. Bowman's capsule, proximal convoluted tubule, loop of Henle, and distal convoluted tubule. Over here, just wrap around the blood vessels around it, and this is what you call as vasa recta. What do you call this as? Vasa recta. Got it? That would work. I didn't make a fancy diagram because, as I know, you do not have much time to see my diagrammatic skills, and also I'm not Picasso. Yes. Now, briefly explain the chemical transmission of impulses. How do you think impulses are getting transmitted out there? Very simple. Listen to me carefully. So there is a neuron. Okay, a neuron is there. Neuron is going to pass the signal. It comes to if at all you have seen the structure of a neuron, it looks like this. So I'll make it and show it to you. Just a second. So I'm making a very small neuron right over here because, as I said, timing could be an issue. Okay. these are what? These are your dendrites. This is the axon and this is the axon terminal. This is what the cytosine out there. Okay, and okay, yes, and here you have so many receptors on the dendrite. Okay, so listen to me very carefully, and here you have neurotransmitters. So I'll tell you how things work. So suppose there was a neuron before this also. This neuron, that neuron, when it was activated, it released the neurotransmitters. So neurotransmitters were at its tip. The neurotransmitters are going to be released. And when the neurotransmitters are going to be released, my dear children, what is going to be happening? So I can explain it in you two ways, and I'll explain it the second way. Listen, over here, so let's start from here, okay? So there was an impulse came, and because of this impulse, what is going to happen? The neurotransmitters which are there over here, they are going to be released in the space. What do you call this space as? You call this space as synapse. You call this space as synapse. Now the neurotransmitters which are released over here goes and binds on the receptors which are there on what? On the dendrites. On the dendrites. Now when these receptors are active, what is going to happen? There are certain ion channels throughout which is going to get open up. Now when it opens up, ions are going to get inside. Some ions will be moving outside also. This is going to change the membrane potential of the whole neuron, which is why a signal is going to be initiated. An electrical impulse is going to be initiated. So over here you had a chemical impulse being given, right? Now that is being transmitted to a electrical impulse right over here. Why? Because over here the signal came, ion channels opened up, and the membrane potential completely changed. Okay, it completely changed, which is why we say that now it has a new action potential which has been initiated. Same thing will be going up again. It will give information for the release of neurotransmitters, and again it is going to happen in the same way. This is how generally chemical transmission happens in the neuron. Okay, we'll read it out once and we'll see what all things have been missed. Structure. A chemical synapse consists of presynaptic neuron and postsynaptic neuron and a synaptic cleft. So this is the synaptic cleft. So listen to me. This, before the synapse, whatever knobs you have, you call this as presynaptic knobs. After that, whatever neuron you have, these knobs are known as presynaptic knobs. Okay, these knobs are known as presynaptic knobs, and these presynaptic knobs belong to the presynaptic neuron. That is, before the synapse, what neuron is there is known as presynaptic neuron. Next neuron which is up there, it is known as postsynaptic neuron. That's all. Done. Okay. Now when an action potential reaches the axon terminal of the presynaptic neuron, it triggers the release of neurotransmitter. Now when the action potential is going to reach the knobs, okay, of the presynaptic neuron, what is going to happen, my dear children? It triggers the release of neurotransmitters. So neurotransmitters are being released. Now, synaptic vesicles move towards the membrane, fuse with it, and release neurotransmitters into the synaptic cleft. So there will be some vesicles given out over here, and my dear children, that is a whole process, okay? And those vesicles which are there will be fusing with the membrane and releasing the neurotransmitters into the synaptic cleft. Okay, now binding to receptors. The neurotransmitters diffuse across the cleft and bind to the specific receptors on the postsynaptic membrane. So in the postsynaptic membrane now, the neurotransmitters which are going to be released are going to bind on the receptors on the dendrites. Now this binding opens the ion channels, allowing Na, that is sodium, and other ions to enter, changing the membrane potential. That is going to be changed. As I told you, when it binds, the ion channels are going to open up. Now impulse transmission. The change in potential generates a new nerve impulse in the postsynaptic neuron. The next neuron, it will create a new impulse, and that is how it is going to go. Now listen, it can be an inhibitory signal, it can be an excitatory signal also. Either ways that can happen. Okay, yes. So that means it can either inhibit or promote later on. So this is a general idea that you should be having regarding the chemical transmission of impulses. Done. So yes. Is there anything else? No. So I think now we can take a break and go ahead and read all the very important things that have been taught. Don't take too much of your time. Just go through it and come back again. Done. And I'll also be back. Okay, so I hope we are ready with part D. That is a 5-marker question. You have to answer any 4, but we will attempt all of them. And my dear children, we'll do a real quick revision of whatever is given over here. Okay, done? Done. Okay. The first thing is enumerate the salient features of pteridophytes. You know what? Pteridophytes are pretty famous for one very important thing. That is, this is the first division of plants, okay, or the first type of plants who were having the vascular system, that is basically the xylem and phloem for the transport of water as well as plants. So they were the first vascular plants. They are the first land plants to develop vascular tissues such as xylem and phloem, allowing efficient transport of what? Water as well as minerals. Now where do they live? They live usually in cool, damp as well as shady places, but that does not mean that they cannot stay elsewhere. They also sometimes grow on sandy soils also. Okay, so cool, damp and shady places, and though some can grow in the sandy soils as well. Now, sporophyte is dominant. So, sporophyte stage of a plant is a diploid stage. I say that there is 2n number of chromosomes up there. Okay, and this stage is dominant in this particular plant, that is pteridophytes. So unlike bryophytes, which was just before them, so from bryophytes they evolved into pteridophytes, my dear children. So bryophytes were the initial one, and they had gametophyte stage as the dominant one. But in pteridophytes, the sporophyte, that is diploid, is the main plant body. So all the cells over here are going to be diploid in nature, which is differentiated into true root, stem as well as leaf. So this sporophyte stage is a proper thing. It has the root, it has the stem, it has the leaves as well. But one very important thing, sporangia and sporophylls. So spores are produced inside the sporangia, which are borne on specialized leaf-like structure called sporophylls. Now when I say that this is a sporophyte, okay, when I say that this particular plant is sporophyte, you will be able to spot leaf-like structures which are known as sporophyll. And you know what? On the sporophylls you will be seeing sporangia. What do you find? You will be finding sporangia. Inside the sporangia, what do you have? You have spores, my dear children. What do you have? You have spores. Spores. And this is what is found in Selaginella, Equisetum and things like that. Sporophylls form strobili as well as cones. So as of now you saw leaf-like thing, right? That was happening in Selaginella as well as Equisetum. But in the case of other plants, my dear children, sometimes sporophylls can also form what? Strobili, that is cone-like structures out there. Got it? Yes. Next thing, water-dependent fertilization. So basically what is going to happen is that the spores, when they are released, okay, when the spores are being released, they are going to form another structure. They're going to form another structure, and that is known as prothallus. What do you call that as? Prothallus, generally. Okay, and this prothallus would be having organs that will produce the male gametes, organs that will produce the female gametes as well. Now the male gametes which are there, they're motile, they can swim and come towards the female gametes. So over here you need water for fertilization to happen. So basically from sporophyte stage we have shifted to gametophyte stage. How? The spores which were the release, my dear children, they landed up somewhere, and now they are making a gametophyte stage, which you also call as prothallus, and in that you have the male and the female organs. The male produce male gametes and the female organs produce the female gametes. The male gametes will be moving through the water for fertilization. So it is water dependent fertilization. So male gametes, which you call as anthrozoids, need water to swim to the female gamete, that is egg, for fertilization, restricting their spread to moist environments. Okay, that does not allow them to, that is the reason why they are not going to moist areas. Okay, come back. Now, homosporous and heterosporous forms. So there are pteridophytes that produce only one type of spores, and there are, you call them homosporous condition. Okay, now if at all they are producing two types of spores, you call them as heterosporous. Okay, or basically more than one type of spores, you will now call them as heterosporous. So some genera like Selaginella and Salmonella produces two types of spores. You call them microspores, which are male, and megaspores, which are female. An important step towards seed evolution. So you have homosporous condition where only one type of spores are being released. You also see heterosporous condition where two types of spores are being released. One is the microspore and one is the megaspore. Microspore means the male ones and the megaspore means the female ones. Where do you find it? You find it in Selaginella as well as Salvinia. All these examples that you see are also important. Okay, going ahead. Now classification of pteridophytes. Pteridophytes basically are classified into four things: Psilopsida, Lycopsida, Spinopsida and Pteridopsida. In that you have examples for all of them. Psilopsida example is Xylotum, and Lycopsida example is Selaginella and Lycopodium, and Spinopsida example is Equisetum, and last but not least Pteroptera example is Dryopteris, Pteris, and also Ediantum. So these are the things that you should be writing about. What? The salient features of pteridophytes, since it is a 5-marker question. At least these points would do. Done. That's all. Okay, next question. List 2 classes of chordates which are oviparous. What do you mean by oviparous? Oviparous means organism that lays egg. Okay, but the question does not end over here. They are asking you, give 2 classes of chordates which are oviparous and also show direct development. What do you mean by direct development? That means if an egg is there, that egg is going to give rise to a new one. It's not like the egg is going to form a larva, pupa, and then it goes. If at all there are multiple stages in between the egg and the adult form, we say it is indirect development. Here we are talking about direct development. So only 2 group of classes are there in the case of chordates, which are both oviparous as well as show direct development. Oviparous, as I said, egg laying. In class of chordates, how many organisms you have? You have multiple organisms, but specifically if I'm talking about vertebrates, okay? In chordata, you have vertebrates. In vertebrates, if at all I talk about organism, you have fishes, you have amphibians, reptiles, as well as mammals and also birds. Now, mammals are not going to lay eggs. Do we lay eggs? No, we do not. We are viviparous in nature. Now, some of the other organisms, they definitely do. But as I said, not all of them show direct development. Only 2 group of classes are there. One is your birds and other one is your reptilia. Birds, you also call them as aves. So class aves and class reptilia, where you have your lizard and things like that, they show oviparous nature and also direct development. So birds also lay egg, reptiles also lay egg. Both of them, the eggs hatches and the young adult is being, a young organism which is just similar to the parent is being born, right? Done. Now, mammals are the highly evolved among animals, right? There are 6 distinguishing features of them. So you have to write 6 distinguishing features, which is why, and that is only how you are going to get the 5 marks out there in total. So the first and the foremost thing is milk-producing glands. So the most unique feature of mammals is what? The presence of mammary glands, which make them mammals, okay, which produces milk to nourish their young. That is the first thing. Body structure. They have 2 pairs of limbs, okay, adapted for different activities like walking, running, climbing, burrowing, swimming, and flying. So it's not just us. You have different group of organisms. You have bats coming under this. You have whale coming under this. You have cheetah coming under this. All of us are mammals, my dear children. And in that, all of them are using their forelimb for different functions out there. Yes? Now we have, that is done. Now skin and external features. Mammals have hair on their skin. Do we have? No, all of us have done waxing, right? No, but the guys will definitely be having. So mammals have hair on their skin and most have external ears, which is known as pinna. This is the external ear, which is known as pinna. It is not, yeah, that's all there. Now, teeth and circulatory system. They have different types of teeth in their jaws. For example, it's not that we just have one type of teeth. If at all you do this, you will be seeing different types of teeth. No, do all of it, you know? Yeah, that's how you do the smiling process, yes. So my dear children, you have canines, you have molars, you have premolars, this and that. So basically multiple different types of teeth are there. That is known as heterodont dentition, okay? Heterodont dentition in your jaws. And their heart is 4-chambered. Our heart, my dear children, is what? 4-chambered. We have 2 atrium and 2 ventricle, and yes, that's all. And why do we have it? To basically make sure that the circulation process is very efficient. Okay, now respiration and body temperature. So mammals breathe through their lungs. Do we? Yes, we do. And are warm-blooded in nature. What do you mean by warm-blooded in nature? Basically, we maintain a constant body temperature. Do we? Yes. So it's like when we go to Manali, it's like our body temperature is going to change? No. Whatever is temperature of the body, it remains the same, my dear children, inside. Because if at all the temperature fluctuation happens, all the bodily functions, normal functions such as enzymes working, everything is going to shut down. So we do not want that to happen, which is why we say we basically breathe in and breathe out and we breathe in more of oxygen. Why? Because this oxygen is required for the production of energy. Why do we require energy now? For maintaining a constant body temperature. So we call these type of organisms as warm-blooded organisms, or you also call them as homeothermous. Okay, homeothermous. So that is the thing about us. Now, reproduction. Mammals have separate sexes. Yes, do we? Male and females are separate. Yeah, and internal fertilization is there. Male and female gametes are going to fuse within the body of the female. So that is internal fertilization, and are mostly viviparous. Yeah, mostly. Okay, it's not like all the mammals are viviparous. There are some exceptions. For example, platypus is not viviparous. Okay, echidna is not viviparous. So you should know that not all mammals are going to be viviparous. So yeah, so some give viviparous means directly giving birth to young ones, and oviparous, you know, right? Yes. So that's it. And the development is going to be direct. There is no larval stage anyways. So this is how it goes. I hope this is clear. Yes. Now the next thing that we have is write the floral characters and the floral formula of family Solanaceae. Okay, so we have to write about Solanaceae. This is the only thing that is given in your 11th book now, with the floral diagram and everything, the formula also. So it's going to be easy, just listen to me carefully, okay? Floral characters. Inflorescence, okay? Solitary, axillary or cymose as in solanum. So listen to me. Solitary means only one flower will be there. That is solitary. Cymose basically means, okay, I'll talk about axillary one. Axillary basically means from the axillary bud you have flowers being forming. That is what you mean by axillary. Now cymose basically means that at the tip also you have flowers, at the lateral side also you have flowers. This is what you mean. So it can be solitary, it can be axillary, or it can be cymose as well. That is the basic thing that you have. Now flowers, if you talk about flowers, they are bisexual in nature. Bisexual means, basically it means that it has both the sexes, that is male as well as female. So male and female part are both there in the case of flower, in the flower. Now they are actinomorphic. What do you mean by actinomorphic? If at all, basically, if you talk about the flower, you can basically cut it any particular plane and it will give rise to equal halves. It will give rise to equal halves, and that is what you mean by actinomorphic. That is what you mean by actinomorphic nature. Okay. Now calyx. Calyx is what? The green color thing you might have seen in flowers, my dear children. Yeah. Below the petals. So that is known as sepal. All the sepals combined together, you will call this structure as what? Calyx. And now we should be looking out over here also. So sepals are 5 in number. Is it 5 in number? Let's count. So from sepal it goes inside. Okay, 1, 2, 3, 4, 5. Okay, 5 of them are there, and all of them are united. How do you know that, ma'am? So basically, if at all, if at all you see over here, you see this means that they are going to be united in nature. Okay, done. They are persistent and show valvate aestivation. What do you mean by valvate aestivation? Sometimes you might have seen aestivation means arrangement of the things out there, right? So sometimes they might be overlapping, right? But here you do not see that. They are connected end to end. Nothing is being overlapping. This type of thing, aestivation, is known as valvate aestivation. Done. So basically calyx, which is there, it is how sepals are 5 of them. They are basically united and basically they have valvate aestivation. Now when we are looking over here, we need to look over here also. See. So I told you they are actinomorphic, that means they can be cut at any direction, so that is represented by this particular thing. Circle and a cross, okay? Bisexual is shown by this, a female and male, both of them. Now calyx is represented by K. Calyx is represented by K, and if at all you see carefully, here it is written 5 and 5 is in that bracket. Why? Because calyx is made by sepals and sepals are 5 of them, and 5 of them, my dear children, they are united, which is why you have a bracket around them. Okay, now the next thing, you're done with the sepals. Now let's go inside, let's see the petals. Petals, my dear children, a group of them, you call them as corolla. How many petals do you have? Again, let's see, 1, 2, 3, 4, 5. We have 5 of them. If at all you look them carefully, again they have united connection. Okay, they are united to each other. And do they overlap? No, they are not overlapping. They do not have any other structure. It's the same thing happening up over here. So they also show valvate aestivation. Okay, so first of all, how do you represent that? How do you represent corolla? Corolla is represented by C. Now how many petals are there? 5 of them. So C5, and the 5 of them are unified. So C5 and then put a bracket. Got it? Not over C but only over 5. So that is how it will look. Just a second. This is how it is going to look. This is a bracket also. Now, next thing, what do we have? We have androecium. Androecium is the male sexual part. That is basically the stamen. So, stamens are 5 of them in number. Oh, is it? Yes. 1, 2, 3, 4, 5. 5 of them. And there's a condition that is being mentioned, and that is known as epipetalis condition. Where is it? Yeah, epipetalis condition. What do you mean by that? The stamen, my dear children, are attached to petals. You have never seen that, right? Very, very rare. You might not have much seen that. Chess. Have not seen that much. Yes. So on the petals you have basically the stamen being attached. It is not attached anywhere else. So this is known as epipetalis condition. Okay, so that means the corolla which is there and the stamen which is there, they have some connection, right? Yes. Or the corolla and the androecium has some connection, right? Yes, ma'am. So androecium, my dear children, we learned there are 5 stamens, so you put 5. And since they both have connection, we put a line like this. That is what do you mean to show? Okay, you are trying to show what? It is epipetalis condition. Done. Okay. So until androecium, it is being done. Now what do you have? You have gynoecium. Gynoecium is bicarpellary. That means you have 2 carpels. Okay. You have 2 carpels, as in one ovary, one stigma style ovary, again one stigma style ovary. So 2 carpels are there. So it can be like this. Now, these two carpels which are there, they can be united also. Is that seen over here? Yes. And that is mentioned by syncarpous condition. So basically, you just have the stigma and style, but the ovary is united over here. Two carpels are there, but they have united ovary. So this is known as syncarpous condition. Okay, and the ovary is superior. What do you mean by that? So if at all you have seen the diagram, if at all you have seen the flower, above everything you have the ovary being resting. The ovary will be resting above everything. This is what you mean by superior ovary. Superior ovary. Now inside the ovary, my dear children, what do you have? You have a space that is known as ovarian cavity. You call this as locule also. So how many locules does it have? Over here, if at all you see this, the locules are two, bilocular. This is one part of the cavity, this is another part of the cavity, which is why you call it bilocular condition. Okay, bilocular condition. Inside the ovary, what do you have, my dear children? You have ovules, and ovules are so many. These all things that you see over here are ovules. So ovary is there, ovule is there. Do you think ovules are flying and staying there? No. You need some support, and that support is provided by placenta. And depending on the positioning of placenta, you have different type of placentation. Over here, the placenta is in the middle, and basically it is forming ridge. It is forming a ridge in a way that the locule which is there, it is divided into two parts. So basically this type of placentation, where it is in the middle and it is forming ridges out there and all the ovules are being supported by that, you call this as axile placentation. What do you call this? Axile placentation. So that is also being seen over here. It is axile placentation. So bicarpellary and syncarpous, ovary is superior, it's bilocular, and placenta swollen with many ovules, and it shows what? Axile placentation. Done. Now what about the fruits? So over here, how do you show that? First of all, so if at all you see this, G stands for gynoecium, that is the female part. Yeah, and I told you it is superior ovary. That means it is above all. So which is why you make a dash under the gynoecium. You make a dash under the gynoecium. That means that it is superior ovary. Now how many, what do you say, okay, just a second. I told you it is bicarpellary in condition, right? I told you it's bicarpellary in condition, which is why it is 2. And they are syncarpous, the carpels are being fused, which is why you put it in bracket. Okay, the 2 is in bracket. Only the androgen, the stamen, is not in bracket, because why? Over here you can see they are not basically fused to each other. They are not fused to each other. Yeah, the fruits that they form is either in the form of berry or capsule. The seeds are going to be many. All the ovules are going to transform into the seed, right? And my dear children, always remember there will be little amount of endosperm still left back. Okay, it is endospermous. So there will be endosperm which is there. Then this is a thing, please know about the full formula. Only Solanaceae family is there for you to remember in that way. So yes, okay, done. Now, given below are the sketches of cell A and cell B. Which one of these is a plant cell, and give reason and support for your answer. Which one do you think is a plant cell? I can clearly see that there is an extra wall up here, and also there is a very big vacuole. And this is a distinguishing feature of what? Plant cell. So obviously B is a plant cell. Now list the cell structures which are common to both the cell types. So most of the cell structures are pretty much common except a few. What are those few things? Do you think we have chloroplast, my dear children? No, right? Animals do not have chloroplast. Otherwise we also would have stood under the sun and do photosynthesis, right? We do not have chloroplast. That means we do not have plastids. Okay. Do we have big vacuoles out there? No, we have small vacuoles out there. Here there is very big central vacuole and all things like that, right? Apart from it, what is something that is, okay, you have been asked what is common, right? And I'm talking about what is not common. So I directly jump into this thing. Name the structures found only in animal cell. Okay, so just a second. We'll talk about what's common. That is even more better. Okay, so let's see. So cell membrane is common, cytoplasm is common, nucleus is there, mitochondria is there, ribosome is there, endoplasmic reticulum is there, smooth and rough, both of them, okay, ER. And Golgi apparatus in animals and dictyosome in plants. So basically in plants also you have Golgi apparatus, but it is specialized form, you call this as dictyosomes. But anyway, these all things are common. Now you have been asked one more thing, the structure that is found only in animal cell. So you might have heard about centrosome, I told you, right? Centrosome has centrioles. Centrioles give spindle fibers, which is there for cell division. In the case of animal cells, you have it. In the case of plant cells, you do not have it. So how do they do that? They have some other things for helping them with the process of cell division. But they do not. Plant cells do not have, my dear children, centrosomes or centrioles. I hope that's clear. Yes. Okay, now over here when I said that it was plant cell have a cell wall made up of cellulose. Distinguishing features you have to tell, okay? Even though if you're not able to see it, you just mention it. Large vacuole for storing water and chloroplast for photosynthesis, which are absent in what? Animal cells. All these things are absent in animal cells. Please mention that also while you are saying that plant A is plant B is what? Basically a plant cell. The picture B is basically a plant cell. Cool? Now, what do we have? Next question. Give the schematic representation of glycolysis. Glycolysis means what? Breakdown of glucose to pyruvate. A glucose is a 6-carbon compound. How many carbons are there? 6 carbon compound. The pyruvate, on the other hand, my dear children, it's just a 3-carbon compound. So 6 was there. If we divide into half, you get 3 pyruvate, right? But you get one more 3 because 3 plus 3, that's how 6 you will get. So basically, by the breakdown of 1 glucose, you get 2 pyruvate molecules. 2 pyruvate molecules, each 3 carbon each. So how do you show that? So first of all, glucose, which is 6-carbon compound, you convert into glucose 6-phosphate. Glucose 6-phosphate. So you have spent some energy over here with the form of ATP, which is why you have got a phosphate group to be added up here. So the 6th position you have the phosphate group. So glucose 6-phosphate. Now you have to make it more reactive, which is why you convert into fructose 6-phosphate. So glucose 6-phosphate is now converted into fructose 6-phosphate. It is 6-carbon compound itself. Now what do you do? Again you spend one more ATP, and whenever you are spending ATP, you are seeing one more addition of phosphate. Okay, so it was fructose 6-phosphate, but now at 2 positions you have phosphate, which is why you call it fructose 1,6-biphosphate. So 1 and the first position and the 6th carbon position you would be having phosphate group, which is why you are saying fructose 1,6-biphosphate. Okay, now that is a 6-carbon compound again that you have. Now my dear children, you are going to break the fructose 1,6-bisphosphate into 2 compounds, each of 3 carbons. So basically you get 2 things. One triose phosphate, which is known as glyceraldehyde 3-phosphate. Okay, glyceraldehyde 3-phosphate. Apart from that, you get one more thing, that is dioxyacetone phosphate. And both of them are mentioned as triose phosphate. Why triose phosphate? Because they have 3 carbons and 1 phosphate group attached to it. That is why you call it as triose phosphate. So from fructose 1,6-bisphosphate, you got 2 triose phosphate. One is in the form of glyceraldehyde 3-phosphate and one is in the form of dioxyacetone phosphate. Now you need to understand one thing. Even though divided into 2 things, we do not need that. We need just this. So basically what are we going to do? We are going to make it convert into this. So 1 was already there, 1 is also going to be formed. So in total how many will be there? 2 of them will be there. So 2 triose biphosphate. Okay, 2 triode, actually you call it bisphosphate, not biphosphate, bisphosphate. So over here, can I call this structure now as, just a second, okay? Yes, so basically here itself you got the 2 glyceraldehyde 3-phosphate molecules were there. Those are converted into 1,3-bisphosphoglyceric acid. What do you call it? 1,3-bisphosphoglyceric acid. Now the 1,3-bisphosphoglyceric acid over here again is converted into the other thing, that is 3-phosphoglyceric acid. And how many of them? 2. Okay, 3-phosphoglyceric acid. 3 denotes the positioning, but how many molecules do you have? You have 2 only. You have 2 molecules of this. Okay, got it? Here basically you are making ATP. Until now you were spending ATP, here you are making ATP. Similarly over here, my dear children, you are making NADH as well. Okay, now the 3-phosphoglyceric acid which is there, it is converted into 2-phosphoglycerate. How many molecules? It will remain 2 only. From here onwards, it is 2, 2, 2, 2. Why 2? Because as I said, already 1 3-carbon compound was there. You are making the other one also converted to this. That is why everything is going to be 2, 2, 2, 2, 2, 2. Okay, got it? Now, you have got 2-phosphoglycerate. You convert into phosphoenol pyruvate. Okay, 2 molecules of phosphoenol pyruvate. Now, the 2 molecules of phosphoenol pyruvate is going to convert into pyruvic acid, or you also call this as pyruvate. How many molecules? 2 molecules of pyruvic acid. So, 3-carbon compound you have got. Okay, so that is how it usually goes. And here also, one more molecule of ATP is being made. Then this is how it goes. You have to represent it like this. If you represent it, you will get your 5 marks out there. Yes. Next, joints are essential for all types of movements involving the bony part of the body. Based on this, mention the types and the location in the human body. First thing that we have is fibrous joints. For example, these are joints which you cannot move. For example, here we have joints, okay, and these things, do you think we can move our skull? No. So it does not. Fibrous joints are the ones that does not allow any movement. This type of joint is shown by the flat skull bones, which fuse end to end with the help of dense fibrous connective tissues in the form of sutures to form the cranium. So basically a cranium which is there, it is made up of multiple skull bones are there, and in between these skull bones, what do you have? You have these type of joints, that is fibrous joints out there, which does not allow any movement out there. Now we have cartilaginous joint. Cartilaginous joint basically allows some type of movement. For example, the joints which are there in between a vertebral column. Okay, so the bones involved are joined together with the help of cartilages, and the joint between the adjacent vertebrae in the vertebral column is of this pattern. It permits limited movement. So you can do this, you can do this. Some amount of movement is still possible. And remember over here, these joints which are there, they are joined. The bones which are there over here, they are joined together with the help of what? Cartilage. They are joined with the help of cartilage, and cartilage is something which you can move. That is why you call this as cartilaginous joints. Okay, now we have synovial joints. They are categorized by presence of a fluid-filled synovial cavity between the articulating surface of two bones. So in between two bones, if at all you have a synovial cavity which is filled with fluid, you will now call this joint as what? Synovial joint. Such an arrangement allows considerable movement. Okay, so maximum amount of movement is going to happen in synovial joint itself. Now these joints help in locomotion and many other movements. Okay, now listen, there are different types of synovial joints. You have ball and socket joint, which is over here and here. So humerus is there, and over here, my dear children, you have the pectoral girdle. So there you have the ball and socket joint. So it looks like the humerus end looks like a ball, and there is a socket. So it fits in the socket, and basically it allows the movement of it. That is what you mean by that. Apart from that, you have hinge joint, which is the knee joint. Again, you can stretch like this, and you can make your what do you call the lower part of your leg go up like that? No, you cannot, right? So only until one. I cannot show you over here, otherwise I would have showed you. Only from here to here it can go. It can go back also like this. It can really go back, but it cannot go up like this, right? So that is because you have a hinge joint out there. Now pivot joint is there between the atlas and axis. So atlas and axis is basically the vertebrae that you find in the cervical column. Okay, the cervical column, which is why you are able to do this. Okay, so the joint which is over there is known as pivot joint. Again, you have a limiting movement out there. Now you have gliding joint between the carpals. So in between the carpal is over here, and in between the carpals you have the gliding joint, and my dear children, you have saddle joint between the carpals and the metacarpals of the thumb, which is why you are able to do all of this thing. Okay, so these are few things that are up there. Please remember it, can be asked. Cool. Next thing, Suma is afraid of spiders, is it? Are you? Okay. On sudden encounter with this six-legged animal, she experienced fright and flight emergency condition. Based on this, answer the following question. Name the hormone secreted in response to the emergency condition. Whenever we have a flight or fight response out there, my dear children, you say that in your body, adrenaline and noradrenaline is being released. What is it? Adrenaline and noradrenaline. And which gland is responsible for the secretion of this? Adrenal gland. And adrenal means it is near to a renal thing. What is renal? Our kidneys. So basically the top of our kidneys we have adrenal gland. It's a pair of gland up there. Okay, and this gland is responsible for the release of adrenaline and noradrenaline. Okay, and it is released by, so adrenal glands which are up there, it has outer cortex region and inner medulla region. Within the medulla region, this particular hormones are being formed. Okay. Now describe the effect of these hormones on circulatory and respiratory system of Suma. I'll tell you. So basically the circulatory system effect is that adrenaline when goes up to circulatory system, it makes our heart beat so fast such that enough amount of blood which has oxygen, okay, oxygenated blood can be transmitted to different parts of the body wherever it is needed. For example, our muscle cells. So that is increase of heartbeat. That is why when we see something, we have tak, 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 tak. Because all this blood which is there has to be pumped to different parts of the body wherever it is required. For example, you have a beer in front of you will run, you will need to run, and for running you will need enough amount of energy in your muscle cells, or basically enough amount of oxygen in your muscle cells such that it can do respiration and provide enough amount of energy as well, right? So which is why, suppose there are blood vessels going to your digestive system. As of now, we do not want too much amount of blood go to the digestive system. We want it go to our muscles out there. So what happens over here? Here the blood vessels are going to get constricted. Under the influence of adrenaline and noradrenaline, blood vessels get constricted. You call it vasoconstriction. Okay, and now At the other places such as muscles, my dear children, there the blood vessels are going to get dilated, which you call as vasodilation. What is the use of this? From here, when it is getting concentrated, all the blood which is there, it is going to go a different location, because here it's not very easy to go. You get it? Understood it? So that is the effect on circulatory system. Your heartbeat is increasing, your blood pressure is increasing, and vasoconstriction, vasodilation is seen. Okay, dilation at the muscles and constriction at the places where it do not require much. Now, talking about your respiratory system, there, my dear children, your bronchioles, everything, my dear children, they will be having enough amount of space so that much amount of air can get inside. If at all there is no air, that means no oxygen. If no oxygen, that means in blood, what are you going to circulate? You need oxygen, right? And that is what you are circulating. Oxygen is required for production of energy, which is why you need to breathe also. And for breathing, it makes sure that breathing rate is being increased, and apart from that, there is dilation of bronchioles to allow better airflow into the lungs. So these are the things that you have to write. I have mentioned about everything over here. You can just have a quick snap, and if at all you need a PDF, do let me know in the comment section. Yes, done. Now, the next group of 5-marker question, and you just have to attempt one. Okay, so both starch and cellulose are polysaccharides. That means they are made up of multiple sugars out there. Okay, they are polysaccharides, but iodine gives color test with starch and not with cellulose. So iodine, when it is being put on the starch, it gives the color, that is, it turns into bluish black is what I remember. Yes. Okay, but when you treat it with cellulose, it is not like that. Why? Why is it that? Because the structure of starch and cellulose is different. Starch has a helical structure, whereas cellulose, which is there, it has a linear structure, which is why it is not able to take up the stain. Okay, so one has helical structure, another has a linear structure, which is why it is not able to take the stain. Now, proteins are known as heteropolymers. Why do you call proteins are heteropolymers? First of all, why do you call it as polymer? Polymer means it's something that is made up of small units, that is known as monomers. So monomers comes together to form a polymer. Now I am telling you it's heteropolymer. That means these monomers which are coming together, probably they are not similar. They are different. Okay, they are different. So basically, what is protein made up of? What is a monomer out there? Amino acids. And these amino acids comes in different combination. One can be glycine, other can be lysine, other can be valine, other can be glutamine, anything out there. So which is why you call them as heteropolymer. Got it? Done. Now, presence of malonate inhibits the activity of succinate dehydrogenase enzyme. So, succinate dehydrogenase enzyme is something that is going to convert succinate. Okay, but malonate comes in between and does not allow this to happen. Why? Malonate, my dear children. So, succinate dehydrogenase has a spot where the succinate molecule has to come and attach, right? But at this spot, instead of succinate coming and attaching, malonate comes and attaches, which is why the whole process is not able to take place. So presence of malonate inhibits the activity of succinate dehydrogenase. Why? Because at the active site of this particular enzyme, malonate will come and join. Got it? Yes. Now, why do lipids whose molecular weight do not exceed 800 Dalton come under acid insoluble fraction, that is macromolecular fraction? So basically, lipid which is there, it has very less weight, molecular weight, that is only 800 Dalton, but still you are categorizing it under macromolecule, bigger molecules. Why? Because actually lipids do not stay like that. Lipids basically combine together to form big things. For example, your cell membrane. That is why you consider this as a macromolecular structure. Okay, they do not come as small units, they come together to form big structures, which is why you call it as macromolecular thing. Now, in solutions of different pH, okay, pH, the structure of amino acid changes. Why do you think amino acid changes in different pH? So, amino acids basically will be having two ends. One is your amino end, and the other one is your carboxyl end. Whenever you put in the acidic medium, you put the amino acid in an acidic medium. In the acidic medium, what do you have? You have so much amount of H plus ions. So what is going to happen? Amino group which is there, it will take the H plus ions out there and convert into, just a second, NH3 plus. You get it? You get it? So there is a gain of hydrogen molecule that takes place over here. And when this gain happens, obviously the structure is also going to be changed. Now suppose this is in the case of acid. Now suppose you are putting in the basic medium, the pH is less. Okay, there what is going to happen? The carboxyl which is there, my dear children, that is going to contribute this H. That is going to contribute this H, so it will become COO minus. So there is a loss of hydrogen. And because of this loss of hydrogen, what is going to happen? There will be change in the structure. Got it? Yes. So starch has helical structure that traps iodine, turning blue-black, and cellulose is linear, does not bind iodine, so no color change occurs. Proteins are heteropolymers made up of 20 different amino acids. So we have so many different amino acids. Sometimes it can be 2 different, okay, sometimes few of them will be different, but anyway we have 20 different amino acids, and with that, with the help of that, we make the polymer of what do you call the protein. Now malonate inhibits succinate dehydrogenase. Malonate resembles succinate and competes for the enzyme's active site. I told you, blocking the Krebs cycle. So this is happening in the Krebs cycle or TCA cycle, and it is being blocked there. Now lipids in macromolecular fraction. So though small, they form large aggregates, that is membranes, making them behave like what? Macromolecules. Okay. Now amino acids change structure with pH. So acidic or low pH, it gains hydrogen, becomes positively charged. Basic, high pH, it will be losing hydrogen, becoming negatively charged. This affects the protein structure and also their function. Done? Done. So please do not forget positive charge, negative charge also. Whenever there is gain, positive, loss, negative. Okay. Now study the schematic representation of Calvin cycle and answer the questions that follows. Calvin cycle means what? The dark reaction or the biosynthetic phase of the photosynthesis, right? So what is happening over here? Which is the primary CO2 acceptor? So basically something has to accept the CO2 that the plant has just taken up. What is that something? It is known as ribulose 1,5-bisphosphate. Okay, ribulose 1,5. So it has phosphate at 2 positions. That is why you call it ribulose 1,5-bisphosphate at first position and the 5th position. Okay, so that is going to be what? The CO2 acceptor. So over here, this is ribulose 1,5-bisphosphate. It is going to accept what? The CO2. It is going to accept the CO2. And what does it become? But before that, name the enzyme required for carboxylation. What is the enzyme that is required for this? You require Rubisco. You require Rubisco. What does Rubisco stand for? Again, ribulose 1,5-bisphosphate carboxylase and oxygenase. So over here, carboxylase function is there. Whenever oxygen is going to combine, you will say oxygenase. But anyway, you call this as Rubisco. You call this as Rubisco. Ribulose 1,5-bisphosphate carboxylase oxygenase. Cool, that is being required. Now name the compound formed from the carboxylation. So when ribulose 1,5-bisphosphate as well as carbon dioxide come together, what is the thing that you're gonna get? What is the thing that you're gonna get? What is the name of the thing? It is known as 3-phosphoglycerate, 3-PGA, or you also call it as 3-phosphoglyceric acid. Either way, it's the same. Glyceric acid, glycerate, all the things are same. Okay, now coming back to the topic, how many NADPH molecules are required for synthesis of one glucose molecule in reduction stage? Okay, so listen to me carefully. 1 CO2, okay, is there. In glucose, how many carbon is there? 6 of them. 6 carbon is there. So basically, you need 6 carbon here also from the CO2, right? So usually when you consider just 1 CO2, we say that we need 2 NADPH molecules. We say we need 2 NADPH molecules in one cycle. Now since there are going to be 6 cycles, why? For production of a glucose molecule that has 6 carbon, my dear children, you will say that usually we need a T2. Now since there are 6 cycles, 2 into 6, there will be need of 12 NADPH molecules. There will be a need of 12 NADPH molecules. So NADPH molecules required for one glucose molecule in reduction stage is 12 NADPH for one glucose. Okay, because one glucose is made up of what? One glucose is made up of 6 carbon. You got it? Fine. So yes. So you need 12 NADPH. Now, okay, one more question is there. How many ATP molecules are required to form RUBP, that is ribulose 1,5-bisphosphate, during regeneration? So basically, after everything is being done, it is regenerated. I'll show it over here. So once glyceraldehyde 3-phosphate and everything is being formed over here, we need to regenerate the RUBP. And for that, how many ATP do you require? You see only one. So only one. You are not being asked about different cycles. You have been just asked how many ATPs molecules are required to form RUBP. Okay, you didn't say about when it is forming one glucose molecule. There is no mention about that. So basically you just say 1 ATP is required for this. Done? Done. That's all. Okay. Now the next thing. A heart patient underwent an ECG, echocardiogram. In his ECG report, time period between QRS complex was more than the normal. Okay, based on this, answer the following question. So I'll show you how generally it looks. So this is the QRS complex. Now you are saying that between one QRS and the next QRS, there is too much amount of a time. Is that a problem? Obviously that is a problem. Why is that a problem? Because over here it shows that the heart is beating slowly. It is beating slowly per minute. So you call this condition as what? Bradycardia. You call this as bradycardia. The opposite of it will be known as tachycardia. Okay, so over here, listen to me carefully. The time period between QRS complex represent the interval between the consecutive heartbeats. If this interval is longer than normal, it means the heart is beating more slowly, resulting in a lower heart rate, that is bradycardia. Okay, next question which is there, give suitable explanation for your interpretation. We gave that. What is the importance of QRS complex with respect to the electrical activity of heart? So what does QRS represent? So first of all, you need to know one very important thing. We have the atrium, just a second. Where is it? Yeah, we have the atrium, we have the ventricle. First of all, the blood in the atrium is there. Blood is there in the atrium. The atrium has to contract, right? The atrium has to contract, then only the blood will be squeezed into what? The ventricles. This contraction is known as atrial systole. What do you call this? Atrial systole. And that is shown up by this curve, that is P. That is P. So we have to go one by one. We will not directly go to this question. We'll go one by one. So P stands for what, my dear children? P is the P representing electrical excitation or depolarization of the atria. Whenever we say there is depolarization, that means that basically something is undergoing contraction. So over here, what is undergoing contraction? It is the atria which is undergoing contraction. We say it is atrial depolarization, or we say that it is atrial contraction that is happening. Okay, got it? So actually depolarization happens, which is why contraction happens. So anyways, so P wave represents that. P wave which is there, it is representing that. Fine. So now it has contracted. Fine. Everything is sent to the ventricle. Now the ventricles have to contract also, right? And your QRS wave is going to represent that. QRS complex represents ventricular depolarization. Again, depolarization means what? It leads to contraction of ventricles. So whenever I say ventricular depolarization, that means that it is going to lead to what? The ventricles being contracted and the pumping of blood to different parts of your body and also the lungs and everything out there. That is what you mean by QRS complex, ventricular depolarization or the contraction that leads to what? Contraction of the ventricles. Done? Okay. Now the ventricles are contracted and that is being represented by this. Please know the steps you would be asked to make this diagram also, so please know about that. Then we see there is a T curve, T wave. What is T wave? T wave represents the return of the ventricle from the excited, okay, from the excited to normal state. This is known as repolarization. That means basically they are going to now relax. The ventricles that were contracted, they are now going to relax. That is being represented by the T wave. By the end of T wave by the end of T wave, we say that finally, okay, finally there is an end of systole. That means end of contraction has been officially been done. End of contraction is being done by the end of T wave. Know that. So small information. By the end of T wave, we say that is the end of systole. It is completely relaxed. Got it? That is the thing. So yes, that's all. We have solved all the questions. And my dear children, I hope this video helps you in some ways. If at all you have any doubts, you can ask me. If at all you think that this video had helped you, also please let me know. I'm wishing you all the best. I hope you guys have a great examination. And bye-bye. Take care. Lots of love.